selamat pagi Pak Dr. Dede.
Morning. Thank you very much for inviting.
And thank you for joining us, Pak Dr. Dede. I think we are still waiting for other participants to join. Would it be all right to you?
Okay, no problem. Yes.
Yeah. Thank you. Good morning, everyone. Welcome back to the virtual summer school program. We are now having the 4th day. Hopefully you are still you still have the spirit to join our virtual summer school because today we will be having two speakers. First one is Dr. Dede Heri Yulianto. He is from the National Research and Innovation Agency Indonesia. And in the afternoon session, we will be having Professor Sung Yoon Lee from Kyungpook National University, Korea. So Dr. Dede will be talking about the biomaterial or biocomposite, and Professor Sung Yoon Lee will be talking about the innovation in wastewater treatment. So hopefully you can attend today's lecture from the beginning, from now until the afternoon, as per usual. And I would like also to remind you that the assignment will be due tonight. So make sure that your group already upload the assignment or deposited through the Padlet. You can find the Padlet link on the Telegram. Otherwise, the committee will share you the Padlet link later on the chat. So I think we are now still waiting for other participants to join. We will be waiting for Arwub. Good morning, Priya, Shakira, Prabias. So if you happen to know your friends that are also the summer schools participants, you can also ask your friends to join our summer school. Thank you, Arun. Good morning. Because our first speaker already joins with us in this Zoom meeting. you there, Arum Restu? How's your assignment with your group?
Hello. We haven't made it yet because
All
the
right.
classes will be this morning about biomaterial and biocomposite.
Oh, I see. I
Oh,
see.
so
Mhm.
Bupedia, may I ask several
Mhm.
questions
Yeah, sure.
related to
Please wait a moment. I think the committee will decrease the. All right. Yeah.
okay.
Sure. Mm-hmm.
So we can something. It is a review about. Let's say I have the biomaterial and biocomposite. So
Mm-hmm.
it can be like a review, like a short review, something like that. Or would you like?
Mm-hmm.
Would you prefer to present one paper about biomaterial and biocomposite, or a review of the whole biomaterial and biocomposite?
Oh no, I think you can just choose
One
specific.
paper.
Yeah. Mm-hmm. And actually what is it for, the biomaterial or the biocomposite that you refer to in the paper?
Let's say I have biofilm, something like that. Can I?
Yeah, maybe you can also elaborate with the issue around you. For example, if you want, yeah, for example, if I take the innovation in wastewater treatment, for example, I found in area around my home, like there is a lot of waste from the for the manufacturer or from the plant or something like that, and then um I want to what is it, so yeah, I propose a solution to treat that waste. So so if you can give us the background of why you use this biofilm or what is it for, so it's okay. Just tell us the background and then what is your solution or what is your innovation. If
I
you
see.
refer to one specific paper, yeah, don't forget to mention what is the background and can you find can you find the issue
Yeah.
around your, yeah, around your area.
I think it will be easier for us to like refer to one specific paper and then find another supporting papers, something like
Yeah,
that.
yeah. It's yeah, it's okay. Like I mentioned before, as long as you can give the background and then you can find the same issue in your area.
Yeah.
Uh,
See.
yeah, so it will be okay. Mhm.
Okay. Thank you
And
so
then
much.
yeah, try to give us the yeah, try to explain the solutions or
Yeah.
the innovation or anything. Mhm.
Okay. Thank
I
you,
think
Guru.
it's, yeah, yeah. Thank you, Arun. So yeah, for everyone. So you can always try to Google, for example, or find like what Arum's group did, like find this specific paper and then try to find the related issue in around your area and then explain what is the what is it what is the solution or the innovation that can that you can give to solve the problem. All right, I think is Pamora is here. Mora, are you ready? All right. While we are waiting, I think we please enjoy the video about the biomaterial or biocomposite, and then we will start our lectures today. Please enjoy the video.
Hello, Vidya. Can we start?
Uh, Okay, so our moderator for today's lecture is already here. So I just give this meeting or the to Pamora, please.
All right. Can you hear my voice, by the way?
Yeah, it's clear.
Okay. Okay, good morning, everybody, dear all participants. Good morning to all of us. Thank you so much for attending this virtual summer school, Green Sustainable Chemistry for a Better Future, Day 4. This event is held by Department of Chemistry Education, Universitas Pendidikan Indonesia. Here I am, Abraham Mora. You can call me Mora, who will be your moderator for this day. Honorable attendees, on this fourth day, we will have two interesting topics which will be delivered by two great experts. In the first session, in the morning session, we will discuss about biomaterial, which will be brought to you by Dr. Dede Heri Yulianto from National Research and Innovation Agency. And this topic is covering the SDG number 15 about life on land. Prof. Sung Yun Lee from Kyungpook National University, South Korea, will deliver the second material about innovation in wastewater treatment, and it will be delivered in the afternoon session. And this topic will be cover the SDGs number 6, which is clean water and sanitation. And for your information, all the recorded version of this VSS will be published on the YouTube, our YouTube channel, which is Departemen Pendidikan Kimia. So don't forget to put your comment, click a like button, and also subscribe. Okay, before we begin our session in biomaterial topic, the organized committee will give us a brief video covering the topic of biomaterial. So for committee, please.
Until humans evolved to replace damaged or diseased tissues in their bodies like a salamander, the development of new biomedical materials will be crucial for the well-being of an increasingly aging population. Biomedical materials science is the study of how artificial materials are created for medical application in the human body, for example contact lenses, pacemakers, dental implants, and artificial joints. Millions of patients' lives are transformed every year through the use of biomaterials, giving them better vision, new heart valves, stronger teeth, improved mobility, and much more. Without biomaterials, dentists and surgeons would be unable to perform medical procedures in the way that we're used to. The Biomedical Materials Science degree course at the University of Birmingham combines biomedical science and materials science. Both subject areas are covered equally, allowing students to develop their knowledge and understanding of artificial materials and how they interact with tissues in the body. Graduates of biomedical materials science have successfully progressed in many different careers, including as a medical device specialist, senior scientist, materials engineer, and science teacher at organizations such as Pfizer, Ministry of Defense, and the NHS. Other graduates have continued their studies to master's or PhD level, with some training for clinical professions such as dentistry or medicine. If you want to study a degree program and contribute to improving the quality of life for millions of people, visit our website to find out more.
All right, that is the glimpse video about biomaterial, the topic that will be delivered soon by our speaker. And before we start our session, I will bring you to our first speaker. And before it, let's I read the curriculum vitae of Dr. Dede. All right. Yes, Dr. Dede Heri Yulianto is from BRIN, the National Research and Innovation Agency in Indonesia. He finished his doctoral degree in Ehime University, Japan. After finishing his bachelor and master degree in Bogor Agricultural University and AHIM University, respectively. And his research interest is in the field of environmental chemistry, bioprocess, polymer science, and applied microbiology. In addition to that, in his career, he has published more than 30 international journal papers with H index of 11 and citation about in 235. And here we share to you a list of several papers that have been published by Dr. Dede. For example, I will read for us pre-treated Sjogren's biggest result in more efficient degradation by Streptomyces sps2 in Journal Microbiology Indonesia in 2022. And so many on that cover the expertise in biomaterial by Dr. Dede. All right, any other slide about CV? Okay. All right. Without waiting any longer, so for Dr. Dede, time is yours.
Thank you very much, Mr. Chairman Pak Mora, for
Ayo maju aja biar 6 orang nih, Rapli maju Rapli. Ini biar 6 orang.
11.54 kalau nggak bisa ini.
30 menit gak maju didiskualifikasi ya. Cepat.
Okay, thank you very much, Pak Mora, for the introduction. I would like to thanks for the committee for inviting me to talk this morning related to biomaterial and biocomposites. Actually, if we find terminology of the biomaterials, so we can find three topic related to biomaterials. First is bio-based materials. It means that we develop the material based on the biology and also bioactive material. These materials are still active during have the activity in the biology, and the other is biocompatible materials, as you see in the video previously. So, can you hear my voice, Mr. Chairman? Is it okay?
Yes, all clear,
Okay.
Dr. Dede. Excellent.
Okay, thank you. So, in this morning topic, I would like to talk about the development of microbial-based biomaterial, one kind of the biomaterials for green and sustainable environmental technology. I think this is almost same with the second talk today, but I will mostly talk about microbial-based biomaterial. This is my email, and you can ask or we can correspondence using this email, and we can discuss more about the microbial-based biomaterial developed in green. So, sorry. Okay, so this is our product, biomaterials and biocomposite product, like CBP. This is the composite based on the polymer. So and also bicycle, woody bicycle, and this is the biocomposite panel, and this is the bioconcrete, and this is one of product from the leaf of the plants, yeah. And we also develop biopesticide. This is the microbial-based biocomposite, and this is the house for the earthquake and also bioadhesive. So in this morning, I mostly talk about the how microbial biomaterial product has been developed in green. This product. So the outline of this talk is only three. First is about the Indonesian biodiversity, and we will talk about the fungi, and especially in white throat fungi. And the second is how the biological prospecting and by all we call bioprospecting has been done during these 5 years ago, and also how fungal-based biomaterial application in industry and environment. So we know that Indonesia is the world richest nation in terms of biodiversity, consists made up about 70,000 islands with a unique ecosystem and contain a large number of diverse species. If we more detail, Indonesia has 10% of world flowering species, and also about the 12% of the mammals and 16% of reptiles, and also 17% of species bird. And how about the fungi? So the number of the fungi in the world is estimated around 1.5 million until 3 million. And about 6% is estimated around in Indonesia, about 86,000 species. And do you know that this number is only has been identified around 1.6% from this 86,000 species. So it is very, very small research or identification of the fungi in Indonesia, but we are the large number of fungal diversity. And mushroom, if we talk about the mushroom of fungi, it consists several family or yeah, like Ascomycetes and also Basidiomycetes. In this talk, I will mostly discuss about the Basidiomycetes fungus. So especially in white throat fungi. White throat fungi usually grows in the dead wood or rotting wood. Why? Because this fungi can use lignin content in the wood to metabolize for their growing. So they use lignin uh, by using the ligninolytic enzyme system, such as lacase, manganese peroxidase, and also some cellulase and silanase enzyme. So if we look more detail about the woody composition, we can describe like this. This is the cellulose fiber, and this is the hemicellulose. It link between the cellulose and lignin. This is the lignin. And this white throat fungi mostly degraded lignin by using this ligninolytic enzyme system. So why lignocellulose is a very important? For example, Indonesian has so many biomass, and this biomass consists of lignocellulose material. This lignocellulosic material consists of, if we more detail, this is the microfibril, and this is the cellulose chains and the cellulose molecule. And if we hydrolysis, this become the glucose or more monomer sugar. The other is semicellulose material, consists about 20 until 30%, And this is the lignin. Lignin consists 15 until 25% and extractive. So mostly the white fungi using this lignin to eat it and become the metabolism grows. So some novel application of fungi, uh, there are many research about the bioremediation and a very limited research about the micro leather. I will discuss this product, micro brick, biofuel, pulp and paper industry, etc. And also this white fungi can produce enzyme, and we also research about the how this enzyme produced by this white fungi, and we apply this lacase for many purposes. So one important step for collecting the mushroom or fungi, we call bioprospecting. Bioprospecting is the systematic search for genes, natural compound, design, and world organism in wildlife with a potential for product development by biological observation, biophysical, biochemical, and genetic method without disruption to nature. So this is the step for the bioprospecting. First, we collect a sample. The second, we isolate and screening for potential white-hot fungi, gene, or enzyme, and we identify it molecularly, and we develop the product and testing and dissemination and commercialization. So in the first step, we already conducted isolation and collection from many other location in Indonesia, from Aceh to Merauke. So we collect all these uh, fungal mushroom and from several, for example, this is we already involved in the Erwin uh, exploration 2016 to Gandang Dewata Mountain in West Celebes, Sulawesi, and also in Taman Nasional Laiwangi Wangameti in Sumba Island. And also we collected from Taman Eden in Toba and Taman Nasional Berbak Sembilang in Palembang, Jambi, and also other program. So I just only talk about some example of our collection. For example, this is we collected from Toba Samosir Island. In this place, we collected about 175 mushroom sample. And in Jambi and Palembang, this is the a very different kind of region, like mangrove, black water, and other is forest. So from this area, we collect about 108 sample mushroom. So why thought fungi? uh, we collect white thought fungi because we would like to use this fungi for the bioremediation of textile dyes. So why white thought fungi can degrade dyes? So as I previously mentioned that this fungi can degrade lignin using the ligninolytic enzyme system, and the lignin structure is mostly phenolic compound, and also the ligninolytic enzyme such as lacase has the very wide substrate, so it can use to degrade also the compound phenolic compound, especially dyes. So if we put this part of the mushroom into the center of the agar medium containing the dyes, and for the several days, 3 until 7 days, they can decolorize the dyes. Here, to become decolorized, and it means that the fungi have the ability to degrade the dye's color in the media. So therefore, we isolate and screen this potential fungi for wastewater treatment. So we collect all the fungi using the selective agar media. Usually, they use Zapekdoc containing lignin agar media. If the fungi, this is the selective media for white throat fungi. So other fungi difficult grow to this medium. So we only use this selective agar medium. Then we isolate. Then we use this fungi to screen in the dye compound. Usually, the scientists use this LBBR dye, Temazole Brilliant Blue R, to screen the ability of the fungus to degrade the dyes. So if the fungi positively active, they can decolorize the LBBR to become colorless, and we isolate this and purification, and we will obtain the potential fungi to degrade the wastewater. So during the screening of the potential fungi for textile wastewater treatment, we use two methods. Like we observe the diameter growth of the fungus, and also we observe the decolorization diameter during the growth for 1 until 7 days. So usually the growth is faster compared than the decolorization rate, but some fungi will decolorize higher compared than the growth itself. So from this method, we then calculated the growth rate. The unit is centimeter per day, and the decolorization rate unit the day centimeter per day, and we select the best fungi. For example, like this. So after we identify the fungi from several places, we calculate the growth rate of the fungus. For example, the Tomates Hisuta D7, 1.34 centimeter per day. It means that every day, these fungi can grow around 1.34 centimeter. And it can decolorize the dyes every day 1.42 centimeter. So from this calculation, we can select the very, very potential fungus, a very high decolorization rate of the fungus. So this is the like a method for fungal species identification. I think this is the biological approach how we compare by using the mycelia and also the mushroom itself to getting the species name confirmation. So as we know that in Indonesia, around 85% river in Indonesia has been already contaminated. So I take this photo in several places in Central Java, and I also collect from the river so many direct contamination from the textile. For example, batik industrial. Like this is the red color, this is the green color, sometimes purple color, sometimes black color, and also the odor is the smell is not good. And these dyes usually contain some toxic compound, like this is the anthraquinone compound. Yeah, this it is a chemical, and also azo, and this it means that diazodiaz. This is one azodiaz, monoazo. This what is it?
Yeah.
It's a reactive dias, and also this is a monoazo dias, and this is also monoazo dias. So every year, around 2,200 and 1,000 tons of dye expelled into the global environment. So, and this is a very recalcitrant and difficult to degrade because the azo dyes is prepared for the very good dyes. Therefore, our textile cannot be easily degraded by the photo oxidation in the sun or by washing. So it is resistant to aerobic wastewater treatment, and it can be environmental problem for us. So some textile dyes is irritated to the skin and eye. We can find around 400 dye compound can be irritated to skin and eye. And the other is mutagenic or carcinogenics. This is a very dangerous for our skin if we everyday contact with these dyes. Uh, so how this textile dyes wastewater treated? So it can be using the conventional technique and advanced technique. So we can divide it into three, like a physical method, like using absorption, membrane filtration, coagulation, and flocculation. Chemical methods using the advanced oxidation and ozonization, and biological methods using the enzyme and microorganism. In the physical methods, using the absorption, the dye will absorb into the material. Therefore, we work to how to prepare this absorption material, a very high maximum absorptivity of the material into the dyes. And using the membrane filtration and how we could filtrate the dyes. Therefore, only the water can release and the dyes stop in the membrane filtration. By the flocculation and coagulation, this is the pollutant dyes. We added the coagulant, for example, and the dye will precipitate in the bottom or sometimes in the upper area. By using the chemical methods, for example, this is only for example, the researcher Carter et al. 2018 until now, 2022, they already developed the zero valent aluminum. So this zero valent aluminum will oxidize some contaminant and remove the COD until 94% and color removal until 98% and ammonia removal until 58%. By using the ozonization, we use this ozone to oxidize the pollutant and become the oxidized product and produce the oxygen. This is using the catalyst sometimes. Or sometimes they're using also the penton reaction to degrade the pollutants. In terms of biological methods, we use the fungi or bacteria to detoxify the wastewater, or sometimes we also use their enzyme to detoxify and degrade the wastewater. This is a very simple method. And every strategy has the limitation. For example, chemical method uh, has the advantage like a simple, a rapid, efficient process, and also multiple approach. But it can be mainly used only in the laboratory scale and economically not feasible for small, medium entrepreneurs. Yeah. For using the membrane filtration, it is also rapid, straightforward, and suitable for almost all type of diets, but high energy, using high energy. Sometimes not feasible for small, medium enterprise, yeah, because can make a clogging using the filtration, and we have to remove and change the membrane filtration maybe for every day or 2 days, 2 days every 2 days. Yeah. Using it for coagulation and flocculation, also it is a very simple method because we just added the flocculant and we shake for stir for only 1 or 2 minutes, and then the dyes will flocculate it, will precipitate it. But sometimes the disadvantage is sludge generation. Yeah, and this method is only like move the pollutant into the other uh, condition. Yeah, so it will increase the process cost. So I also have developed some coagulation and flocculation, but very sorry, this video cannot, I don't know why cannot be played because very big video. This is a very simple method. Only 1 minute we can decolorize and precipitate the pollutant here, and the flock will be stayed in the bottom. Sometimes in the bottom, sometimes in the upper layer. But it is a very simple process. But we have another pollution like this. How we treated this flock after we remove the color from the water. So okay. Oh, sorry. It's very big file. Oh.
Would you like to share us a video, Dr. Dede?
Yeah, maybe later I will share the video because in this PPT it's very, very,
Okay.
yeah, heavy.
Sure.
Later I will if we have time. Okay, I will continue. But okay, so according to my experience, how the efficient treatment methods. First, I saw that absorption method, like using the PC absorption or chemisorption, is a very easy, simple, flexible, operative, insensitive to noxious material. I think almost material can be absorbed by using our material, economically feasible and methods for wastewater treatment. So we can develop low-cost absorbent, nano absorbent, or biosorbent. Yeah. So low-cost absorbent like a natural agriculture waste, industrial waste using the zeolite, chitin, clay, lignocellulose material, and others. And nano sorbent usually carbon-based nanomaterial, but the other material like a nano zeolite, yeah, and nanoparticle ferro nanoparticles. Also, it is a hollow site nanotube can be developed to make a nanoabsorbent. Or not biosorbent can be derived from the biological source like a microorganism. So in this term of absorption, we should measure the absorption capacity, maximum removal efficiency, and it is a very ready, available, and cost-effective and non-toxic material. So the other efficient method is a biological method. Yeah. So microbial biodegradation, it is a simple and economically attractive technique and can degrade toxic compound into non-toxic compound. The other method is only remove the pollutant to only move to other place like sorption and flocculation and other. But using the microbial biodegradation, the toxic compound can be degraded into non-toxic compound. But the disadvantage is this process is very slow. usually 6 until 30 days. So in this term, we can develop how to find the super microbe or how to develop super microbe by using the recombinant protein and others. And the other is we immobilize or we combine this microbe with the absorbent or super absorbent. So the process will be uh, simultaneously between absorption and biodegradation. And this is the enzymatic biodegradation, also can detoxify the toxic compound into non-toxic compound. This is the process is very fast or in hours, and it is challenging because the production of the enzyme sometimes a very high cost. So how we challenge to find the novel enzyme and to develop the recombinant enzyme and to immobilize or combination enzyme with the absorbent. So uh, based on my experience, combination of biological and physical method is a efficient treatment. In this term, we can analyze the immobilization yield, absorption capacity, maximum decoloration efficiency, usability, stability, and also degradation product analysis and toxicity assessment. So in terms of this, I developed microbial-based biomaterial for textile dye wastewater treatment. This is the combination of biological and physical method. So we have already isolate, screening, and identify the potential fungi. We also already degraded the dye in the liquid medium using the microorganism, and we also already produce the only enzyme, and we use this enzyme for the decolorization and degradation directly using the enzyme. And this is the key technology, how we develop microbial-based biomaterials using the fungal mycelial immobilization and also enzyme immobilization on the biomaterials. And this can use for the pilot application and implementation and application in the real wastewater treatment. Why? This is the key technology because the protein biomaterial or microbial biomaterial over the stability, yeah, offers the stability, reuse stability, operational stability, and also storage stability. Storage stability means after we produce this product, we can store in the, for example, very cool room or in the storage room, and also the operational stability cannot be influenced by the temperature or pH, yeah, and also usability is mean can be used, can be reused for several times, yeah, and also stability during the reaction. So this is the decolorization of the textile dyes by the fungal mycelia. This is only for one example of the decolorization, how we decolorize the dyes using the fungal mycelia. We use several type of dyes like anthraquinone dyes and also these azo dyes, and we see that the selected fungi can decolorize higher compare other fungus, and we conducting the fungal immobilization. For example, this is, for example, in leca. Leca is like weight expanded clay aggregate. If you see uh, in the place, this is the leca used in the plant. Yeah, I take this photo uh, in Singapore. Yeah, Changi Airport. They put the leca in the airport. Yeah. So after steam activation, yeah, washing, and we culture the fungus, and we obtain this composite, mycelial composite with the leca. We called micro leca. And same analysis of this fungal immobilization, we see that this is the only LECA, and this is the LECA after immobilized with the fungi. So we can see the mycelia can enter into the hull of the LECA, and we use for decolonization, and this is we already registered patent, and it can be used for decolonization and degradation several dyes, anthraquinone, azo, the azo direct, and others dyes. Naphthol, indigosol we already conducted, and we can see here only in 4 hours we can directly degrade it or decolorize the BBR dyes and other dyes. And we also already use this into the textile and batik dye wastewater in several places like in Pekalongan, Purwokerto, and other places. This is the indigo dyes from the Purwokerto, yeah, and this is the from the Jababeka in Jakarta. So this immobilization yield is around 67% equal to 1 milligram of mycelial fungi per gram of leca. Yeah. So we also uh, found that this mycoleca can detoxify the dyes after treatment. For example, this is the 0 hour mean before treatment, and after treated the self viability increase. So we found that the treatment of dyes using this mycoleca can detoxify the dyes itself, yeah, significantly. And we already published in Journal of Hazardous Material last year, 2021, and also can detoxify using the phototoxicity, phytotoxicity using the Vigna radiata and also the Artemia salina. We published this in the Biotech 2021 last year. And we analyzed the metabolic pathway of this anthraquinone dyes. This is my coleca. We apply into the dye and decolorize, and we inject into the LCOB type analysis. We found several metabolites, identify, and we propose the metabolic pathway. For example, this one. So usually the anthraquinone dyes has the parent compound like this. So this one is L1. If we, for example, LB4 site is here, and LBBR is here. So if we treat it using this mycoleca, then this compound will break down into the this one, and the parent compound will degrade it into the this compound. And we analyzes the toxicity and like previous slide. So it is proof that this fungi mycelia composite can degrade the LBBR anthraquinone dyes or AB4 anthraquinone dyes, or the other is AB129 dye into the non-toxic compound. So the other is we use this mycoleca to biodegrade and biodetoxify azo dyes. So we use the diazo dyes. This is a very difficult to degrade it by using the physical and chemical method, but we use fungal mycelial biocomposite to degrade this one, and we analyze this using the UPLC, ultra high resolution mass spectroscopy, and we analyze the biodegradation pathway. We also compare using the molecular docking and cytotoxicity and genotoxicity, still under review in chamber spare. So this is the result of the project. So this is LB5 dye. We analyzes the all these peak, and we found that this is the 0 hour, and after reaction the LB5 dye is removed directly, and several peaks are detected in the spectrum. So we detect that this LB5 dye, many compound detected in the LC orbit trap. I mean this is using the FTICRMS analysis in Kyunpok National University using their equipment. And also this one is AB113 dye. This is the azo diyes, and we see that this peak is significantly removed after the degradation, and several peaks also found in the metabolic pathway. And this is the acid orange 7. It is a very difficult compare other dyes, although only one monoazodides, because we this paper is understanding the biodegradation pathway. By using this molecular docking, we found that this enzyme, the lactase enzyme, is difficult to degrade this acid orange 7. And this this is the reusability. The most important factor for efficient method. After several times use, this microleca still higher decolorization ability. And the enzymatic activity still, the lactase activity still detected. It means that this fungi very good immobilized in the leca, so that the reusability is high, and also the lactase activity is very still can be detected during the decolorization process. So we also applied the microleca into batik wastewater treatment in Pekalongan. So I go to the field laboratory, so I cannot bring the spectrophotometer data, but I only use the light here. Before treated, the light is very dark, and after treatment to 24 hours, the light is a very uh, can be seen diameter like is higher compared than before treatment. Then this is the color after, before and after. And after we applied for 3 weeks, and the owner sent me the photo, and this is the 0 week, and this is the 3 week. The color is removed, and the wastewater can be removed by this microleca. And also the pH decrease into the 8, and the COD level also decrease, the decolorization also decrease. And we check in this wastewater whether this is the microleca, whether the lactase activity detected or not in this wastewater treatment. Because I didn't bring the equipment, I only used the color change using the ABTS. If the lactase inside into the wastewater, then the color will change into the dark and with into the green color. It means that the enzymatics detected in this reaction. So this is the application of microleca in the textile wastewater. It is in the Jababeka in Jakarta, and we already published this paper. The decolorization yield around 74%, and the COD level decrease, and other. So how we use enzyme now? So we also use this enzyme directly, not the mycelia. So we biodegradation and biodetoxification of textile dye wastewater using immobilized lactase enzyme. So this fungi produce the lactase enzyme. So how we collect, we isolate the lactase. So first we fermented the fungi into the lignocellulose material. Like what I already mentioned before, that this fungi grows optimally in the lignocellulose material. So we incubate it in the 1 hour, 1 month. After 1 month, the fungi extract, I mean the enzyme extract it using the homogenizer and buffer, centrifugation, using the ammonium sulfate precipitation, and also lyophilization, and the other technique for purification. So we obtain the crude powder. So this crude powder we use for the enzymatic decolorization directly during several hours. Yeah, only enzyme. So we obtain the maximum from the zero point 0.1 until zero point 0.2 unit per ml per milliliter. And all these enzyme can decolorize dyes depend on the structure of dyes itself. Yeah. So for example, we use several dyes, LBBR, AB, acid blue 25, acid blue 113, acid blue 129, and the others. Mostly all dyes can decolorize by the lacase from Thymetes hirsuta, yeah, our fungi. But some dye like AO7 is very difficult, as I already see. So this is the lacase immobilization. Why we need to use lacase immobilization? Same, we need to increase the stability, use stability, storage stability, and also operational stability. So we already developed like using the calcium alginate, polyphenyl calcium alginate, modified silan, modified perlite, and also modified lica. So this is the how we improve the how we use lacase immobilization in calcium alginate using the sodium alginate, calcium chloride, and we will get that like a small ball. Inside the ball is the crude enzyme, and this is the like this. This is the ball, and this is the enzyme. It can be used like this. This is the, for example, this is the wastewater. We apply into here and directly here, or we use from bottom to the top. We analyze this here every hour. So we obtain that the only 2 hour, the enzyme can decolorize the dyes with detoxification, yeah, after treated. Uh, the other is we can use a polyphenyl alcohol, polyphenyl alcohol calcium alginate bead. This is the bead, yeah. The bead is become white color. It's almost same, but it is using the double layer. We optimize the boric acid concentration. We obtain the 7% of boric acid concentration, and we compare the calcium alginate with the polyphenyl calcium alginate. And the polyphenyl calcium alginate improve the sorption capacity, decolorization, strengthen, and also reduce the enzyme leaking. This is very important. The enzyme should be reduced. The leaking enzyme should be reduced after immobilization. Yeah. And this is the silan. Silan obtained from the pulp and paper wastewater. We added the citric acid in 150 degrees Celsius, and we obtain the modified silan. And this is the citrate silan. If we analyze using the FTIR, the sealant is here, but the citrate sealant can be can add one peak showing the CO staging. This is it means the carboxylic acid, and we use the modified sealant using the PPA, yeah, polyphenyl alcohol, and the enzyme lacase using the peristaltic pump. And this is the reaction, almost same with the previous one. And we obtain the optimum pH can use in from 6 to 10 at 40 degrees Celsius, usability around 6 cycles, and dye decolorization of 57% using the AB5 dyes. Yeah, this is in Bangkari, our partner. And also we also using the perlite, lacase immobilization perlite using the APTS, yeah, and glutaldehyde. We obtain this immobilization. We use for the decolorization of dyes using these dyes. And this is the enzyme, almost same with the mycelial immobilization, but this is the enzyme immobilization. So we published this 3 biotech. So this is the only LECA, and this is the LECA with enzyme. And this is the only LECA after treated into the naphthol dyes. So we can see that the naphthol dyes absorb in the leca. But after application using the uh, enzyme immobilized in leca, there are no color in the composite here. So this is the reaction how we develop leca and the leca lacase. Yeah. So according to FTA analysis, this is the enzyme only. Yeah, we can see many peak for the amide. This is the amide spectrum, and this is the leca. And after addition of enzyme into the leca, we can see many different peak. So it means that the covalent immobilization has been developed during this immobilization. Okay, the other is we use the fungi for the bioremediation of oil, yeah, and we detected also the metabolite analysis using these fungi. The other is how white cloud fungi degraded expanded polystyrene. This is the polystyrene structure, and this is the application of polystyrene. We have several white cloud fungi already isolated from the other place in Indonesia, and we biodegraded this polystyrene for 1 month. Actually, still running until now. And we analyzes the microstructure degradation of FPA. This is the control. And after addition of the fungi, mycelial fungi can penetrate the surface of the APS and making the microstructure degradation. And also after the FTIR analysis of styrene, this is the polystyrene, we can see that many peaks in the pure APS are removed during the degradation. And we calculated the reduction index of the aromatic group and phenyl group. We can see that BRB11 resulting high reducing index of aromatic group and also phenyl group compare other fungi. But all fungi can reduce the aromatic group and phenyl group from the expanded polystyrene. So also we analyze using the TGA analysis. This is the pure APS. We can see that the what we call the removal of the weight of the APS is faster compared than the only pure APS. You can see here the thermal analysis show that the like here the TG analysis decrease around 1 until 2 degrees Celsius. And also we use a pyrolysis GCMS analysis. This is pure APS, and this is the treatment by using the fungi. We can see here that BLB11 is so many peaks resulting as the biodegradation result after the APS degradation. So we conclude that these fungi potentially degraded the APS during the incubation. So the other for using the mycelia is to develop mycoleather. So the mycelia itself is very strong, have the strong ability. So potentially used as a novel leather. So we already developed several fungi and we checked the tensile of the mycelia, and this tensile around 6 until 8 megapascal with the elongation break around 38 until 47%. It is potentially used for the leather. And if you know the Mycotech, Mycobacter Nusantara, we collaborate to produce this leather. And also we also produce microbrick for the structure or ornament, yeah, in the room. And the mycelia itself, especially enzyme, can be used for sustainable biofuels like this. So we produce fungi can directly produce ligninolytic enzyme and also silanase and cellulase enzyme directly. So this already published in Bioresource Technology Report in 2020. After after treated by this fungi, the production of the biofuel is increased dramatically because the simultaneous enzymatic process are involved in the product. So the lacase enzyme from the wild fungi can be used as a green industry. Like this is a technique for application of the lacase. It is our lacase product. We explore exploration and immobilization, recombinant protein, or protein engineering and metagenomic analysis should be involved in the degradation in the experiment. So this is the lacase for green industry, yeah, usually used for bioremediation, pulp and paper, cosmetic, personal care, and pharmaceutical. So lacase enzyme from the white fungi is a very application for many applications, like in the food or in the pulp and paper industry. In many places in pulp and paper industry can be used, like in biopulping, yeah, in biobleaching, sometimes in biografting, bio de-inking, yeah, using the lacase. Uh, Sometimes effluence, yeah, the effluence. This is the effluence of after pulping process can be used for lignin transformation to get the lignin derived chemicals, and we will obtain the green chemicals from the lignin using the lacase enzyme. And also, lacase can be used for novel medical and personal product like skincare. Yeah, sometimes we make IV dermatitis, and the lacase can oxidize the IV compound, yeah, to become non-toxic compound. Or sometimes lacase also can be used in the production of iodine after oxidize this iodate into iodine. Or lacase used in Korea, yeah, in Korea, they use lacase for hair dyes, yeah, and also textile dyes, because after treating by the lacase, the color sometimes become the others very special colors. So they can develop special color from the addition of lacase. And lacase also can be used in the personal care. We already make a collaboration with the one big factory, yeah, enzyme toothpaste to develop this enzyme based personal care. Also, lacase can be used as an anti-cancer. Yeah, this is the research from the Chauhan et al. 2019. This is the total global market of lacase in 2019. Yeah, around 3.1 billion US dollar. Yeah, I think that's all my presentation. But before I close, I will introduce my integrated laboratory of byproduct. So I invited all of you to join with our laboratory. We have so many sophisticated equipment like FSM, microconfocal Raman spectroscopy. I think it is only the best one. In Indonesia, we have 4 laser. Yeah, also for the micro biology assessment, yeah, identification by using the laser without any non-destruction test. Yeah, we can analyze the structure and the chemical component of the sample. And also we have the like a microscope, so many microscope, and FT near, XRD, FTIR, AS, and other component. I think this is the for biocomposite materials. Yeah, UTM, tensiometer, DMI, DSC, FTIR, and also for chemical analysis using the PYGCMS, SPLCGC, and LCMSMS. Yeah. This is for the making the polymer, and this is for enzyme equipment. Yeah, we can purify enzyme using this ACTA purifier. Yeah. I think that's all my presentation. Thank you very much for your kind attention.
All right, thank you so much, Pak Dede, for the nice and excellent presentation. Here, I give you all participant a little resume about what Dr. Dede has been explained. The first one is about biomaterial. Dr. Dede worked with fungi to degrade dyes and wastewater treatment. Begin with isolation and using fungus and using many treatment technique like a physical, chemical, and biological. Especially in biological treatment, he used microbial biodegradation and enzymatic biodegradation for the application, like we have seen before. The application for the microliter and microbrick, as well as biofuels, and as well as lactase enzyme has been applied in the green industry for medical and personal care. Such a great lecture from Dr. Dede, and thank you so much once again. And before we jump into Q&A session, maybe we can give a little rest for Dr. Dede for taking some water maybe. And while we're waiting for a little break, the committee will show us a short video. All right. Thank you so much for the committee. And here we are in the Q&A session. So for all participants who want to ask Dr. Dede about biomaterial, fungi, you can write your question in the AHA slide. Link has been given to you in the chat room, or otherwise you could directly ask the speaker. Please let me know by raising your hand emoticon, and I will let you ask Dr. Dede. So, is there any question from participants who are willing to directly ask Dr. Dede? Otherwise, maybe committee will show us. Okay, Eva Monifah, please, Eva.
Okay, thank you. Please let me not activate the camera because the internet here is not stable.
Sure. Go ahead.
Thank you. Very interesting lecture related to biomaterial, Pak Dede. My question is it possible to use the mineral cellulite repeatedly? The seaweed processing industry into agar-agar today use a membrane filter of cellulite in the filtration process. And this cellulite has a porosity similar to the mineral of zeolite, and the volume is very abundant. About 75% of the total volume in the solid waste from industry of agar-agar. Is it possible to use this cell life as an immobilization membrane? Very interesting and inspiring lecture, Pak Dr. Dede. Thank you very much today.
Okay, thank you, Bu Iva, for the question. Please, Dr. Dede.
Yes, thank you, Mbak Iva. Yeah, yeah, it is very possible. So now I have a collaboration research. We call the Research Collaboration Center for Marine Biomaterials. So we collect uh, bioresources from marine to become a biomaterials. And one of the product is we making a membrane, or the other is we making a biofilms, yeah, from the carrageenan, and we combine with the mycelia, and also we combine with the microfibrill cellulose or nanofibrill cellulose to become the packaging materials. Yeah, we now explore how marine bioresources to become a biomaterials. Yeah, cellulite is a potential for the filtration method, because you, as you already mentioned, it consists of many pores, and it can be used as a absorbent or filtration, yeah, like this. I think it is a very interesting topic, yeah, to collaborate the research together.
Thank you, Pak Dede. I see on the slide that the tools in Pak Dede's lab are quite complete. Well, I hope we can work together on the proposal related to the use of cell like Pak Dede. Thank you.
Yeah, please, welcome. Yeah, we have also so many equipment like BAT, yeah, analysis, and NMR, and yeah, you can use it is open facility, especially when you have contact with researcher from brain, you can access all these equipment. I can help you to analysis using these all equipment.
Thank you, Pak Dede, Mas Abraham.
Okay, thank you, Bu Iva, for the question. Yeah, we know that brain is very equipped, and collaborate with brain is one of the such a privilege, like, for institution like university. Okay, we can move maybe to question in the AHA slide, Pak Dede, because there are so many questions from the active participant, and committee could help me to show the question. All right. Okay, I read it for you, Pak Dede. The first question, from my student, Satria. Satria, could you ask Pak Dede directly, maybe?
Okay, Pak Mora, I can give a question. For Bapak Dede, I have a question. There is a species named fungi protis, or in Indonesian, protista mirip jamur, such as a myxomycota and omycota. Does it has potential for biomaterial as mushroom have or not?
All right, please, Pak Dede.
Okay, I don't know detail about this fungi, but actually the basic concept for making the biomaterials is, first we should identify how we use this fungi for. For example, if we use for the wastewater treatment, for example, dye decolorization agent, then we can starting from the decolorization ability. If we want to use like for the materials with the having high ability, high strength, we can use for using the UTM analysis for the mycelia, yeah, and we we start to isolate and screen these potential fungi from the beginning. And after that, we can develop these fungi for more detail. I think all our biodiversity will have a specific characteristic and specific application for these biomaterials.
Okay, thank you very much, Dr.
All
Dede.
right. Thank you, Satria, for the question. Next question, Pak Dede, from someone named student. I once tried processing batik waste in Malang Regency by coagulation using tawas maybe, aluminum sulfate.
Yeah,
And then it produced quite a lot of residue, and the LC50 value was quite low. The question is whether the residue can be processed with wood root mold, because the pH of batik is very alkaline, around 11.58. Is it possible?
Yeah, actually, I also found what you experience. Yeah, what's your experience? The residue is very quite a lot. So yeah, this is one of the disadvantages of the using the coagulation. Yeah, for example, tawas, and the LC50 value is quite low. You mean low? I see LC50 is toxic, right? The low LC50 analysis concentration is mean low is mean toxic. So yeah, actually we already done this experiment, and white fungi can remove the residue or the flock from the coagulation this material. And yeah, the batik pH, yeah, sometimes 11, sometimes 10, sometimes 9, based on the chemical component in the batik wastewater. But these fungi can grow, yeah, in this pH, because we already use several pH concentration, pH value using the buffer. You know, the buffer is a very stable pH, but the batik wastewater easily decrease pH by the white fungi. For example, in my experiments, the batik wastewater pH decrease after the white fungi application from 10 to around 7 or around 7.5. Yeah, it can be used actually.
Okay, all right. Thank you, Pak Dede. I've been curious about the minimum pH that you use to this experiment for fungi.
Yeah, the water fungi optimum in 4.5 pH, 4.5, because it produce ligninolytic enzyme, and the ligninolytic enzyme, for example, lignin peroxidase enzyme optimum at pH 2, and lacase optimum at pH 5.5, manganese peroxidase optimum at pH 4.5. But sometimes, yeah, by a perspective of this lacase enzyme, we sometimes develop the range of the pH. Some researchers already found that the lacase can be used in the alkaline pH, like a pH 8, yeah, pH 9 sometimes. So it is possible, but usually the lacase is optimum at pH 4.5.
Okay, all right. Thank you, Pak Dede. Next, we move to another question from Wita. This is also our student from UPI. Wita, could you ask Pak Dede directly?
Yes, sir.
Yeah, please.
Dr. Dede, wait.
Tunjukkan aja.
Dr. Dede, what are the challenges of applying Lakas enzyme in Indonesia, and how do you think to handle them? Thank you.
Okay, so yeah, this is a very good question. Yeah, we Brain actually already established how to produce lactase enzyme. As you know that in Indonesia there are no enzyme production. Yeah, so there are so many. There are no industrial, especially produced enzyme. Yeah, so all the enzyme need from the Indonesian industry are coming from China, mainly coming from China. So this is the challenge: how we can establish or build the enzyme factory in Indonesia. Now I try to contact several industry, several company to agree with this. We making or we establish enzyme factory in Indonesia. This is very challenging to applying the enzyme in Indonesia, because you know that our Indonesia is a mega biodiversity, producing many microorganisms and also many enzyme, but we have no enzyme company in Indonesia. All the enzyme needs we import from China and other countries. So this is the challenge. The other is enzyme application, especially in the environment. The environment, we can see that the temperature is high, maybe around 50. So how we produce the thermostable lactase by using the recombinant lactase. And our staff is now already research about this, and he study in Malaysia how to improve the lactase stability in the higher temperature. And also, if we only use lactase powder after we produce lactase and we apply into the river directly, then the lactase will dilute in the water and remove. So the most important is how lactase is covalently, immobilized in the matrix, and we apply, for example, like using the filtration, and the river water will filtrate using this material containing lactase, and the contaminant will oxidize by the lactase, and the lactase itself still recover in the matrix. This is the challenges. How you make the, like we call protein biomaterials. Lactase covalently immobilized into the matrix. Yeah, this is the challenging. How you make a higher reusability of this lactase. We already get the composition, like in the LECA, we can use almost at least 19 cycle after degradation. We use again until 19. The recovery is still 96% degradation. So I can resume. The challenge is now making the enzyme company in Indonesia. Second is finding the lactase biomaterials product, so that the lactase can covalently immobilize in the matrix and the reusability increase. Okay.
All right, that's the answer, Wita. But I just want to ask you, Pak Dede, about how much the lactase enzyme maybe per gram imported from China, let's say.
Okay, so for your information, when the company, local company buy the enzyme from the China, for example, they didn't mention about this is the lacase, this is the glucose oxidase. They only use this is the enzyme. Yeah. And when we check the enzyme data in our, for example, Ministry of uh, Industri ya, atau Menteri Perdagangan ya, Bapenda, you can not more detail about the enzyme imported from other country to Indonesia. But the price is still high. Yeah, I don't know more detail about the price because the company cannot mention it to me. But they usually buy the crude enzyme. Yeah, crude enzyme. Sometimes in the powder, sometimes in the liquid condition. Yeah, mostly in liquid condition. And I already checked from the company, and we Indonesia can produce this enzyme same with the quality with China, for example.
Yeah. So
But if you ask about the pure lactase, the pure lactase is very high, very expensive. I mean, for example, 1 gram is around 2 million or maybe 5 million.
Wow.
Yeah,
Such
1 gram.
huge amount. Yeah. Okay, so after we buy, let's say, crude enzyme, we need to make a purification first before we start to use that.
No need. If you buy the crude enzyme from China, you can directly making the byproduct. Yeah. So
Okay.
yeah, this is the. But the most important is if you want to use this enzyme for food, for example, then the production it should be food
Great.
food grade. Yes, food grade.
Yeah. Okay.
Like
Thank
example,
you.
like example for you, maybe you usually use the enzyme pasta
Yeah.
gigi paste. Yeah,
Yes. Yeah.
it contains at least 3 enzymes there. Yeah, because we develop together with the industry.
All right. Interesting, Pak Dede. Thank you so much. We move to the next question from anonymous. When we precipitation method in dice removal, isn't that mean that dice is still there, just in the bottom and mixed with mud in the water?
Yeah, it is true. But yeah, therefore the precipitation method is not a suitable method for cleaning. Only move the dyes into the precipitated. Yeah.
Okay, next one. Dear Dr. Dede, thanks for a nice sharing. What are the advantages and disadvantages of using bumi as a source of biomaterial compared to other being like bacteria, viruses, or algae?
Okay, so bacteria is fast grow, yeah, rapid grow bacteria. Maybe only in hour. But some bacteria usually produce intracellular enzyme. For example, monooxygenase, deoxygenase, it is intracellular enzyme. But white-out fungi produce extracellular enzyme. So it is easier to isolate, yeah, compared than intracellular enzyme. The other fungi more stable compared than bacteria, but fungi is slow grow compared than bacteria. About the virus and algae, I don't know. Virus usually not used for the bioremediation application. Yeah, but algae sometimes used for the bioremediation application. Almost same. Yeah, algae. Sometimes algae also. Yeah, not so of fast grow compared than bacteria. I think. Yeah. I don't have any experience using the algae.
All
I think no.
right. Yeah. Okay, so the main reason using fungi is they can produce
Enzyme.
extra cellular enzyme that can
Yeah.
easily extracted. All right. Next from UM student again. Yeah. How about SSV, solid state fermentation, sir? Can it be used in the growth of wood root molds so that the extracellular enzyme of the mold are increased for the decolorization process? Please, Pak Dede.
Yes, we produce the enzyme using the solid state fermentation, as I said, by using the lignocellulose material, as you already see in my slide. So yeah, it is a very good strategy for production of the enzyme using the solid state fermentation. And also, if we want to produce the micro leather, as I already present, I also use solid state fermentation. And after this is the uh, very hidden technique. After the mycelia goes into the outer layer of the solid state fermentation, then we open all the plastic and the mycelia will making thick ya, akan menebal, and we slice and we obtain the leather of the mycelia, and then we uh, prepare using the boiling water, yeah, and yeah, etc. We use solid state fermentation.
Okay. To the next question from anonymous. Have you considered using synthetic biology to design a microorganism that can digest the waste that you are targeting?
Yes, of course, synthetic biology is a very good method, yeah, but it is not easy. Uh, yeah, we sometimes want to use the how maybe you already know about the uh, uh, jaring laba-laba, Spider-Man. Yeah, we can develop this jaring laba-laba to become very strong materials. Yeah, very strong materials, and also uh, we can develop most super microbe to degrade waste, especially it can tolerant in the high temperature, high pH, and produce very high enzyme. Yeah, it's very suitable method, but not easy to develop the synthetic biology.
Okay, for your information, all participants jaring laba-laba is spider web. Yeah, spider web. Okay, thank you again, Pak Dede. This is the next question is very interesting. Is there any toxic byproduct that was produced after the treatment of dyes using fungi?
Yeah, we already analyzed using the gene toxicity, using the microbial toxicity, using the plant toxicity. So the byproduct of the wastewater treatment after treated by the fungi is non-toxic compound. It is suitable for application.
Okay, right. So it's safe. It can if used in the environmental, right?
Yeah.
Okay. Let's move to the next question from anonymous. Technically, how do you apply mycoleca to degrade dyes in rivers? Did you just throw it away? Will there be side effects such as disturbing the balance of the ecosystem? Interesting.
Yeah. Very interesting. The Michael Leica can be put in the, for example, uh, in the uh, uh, states, and then the river will to the filtration, and the uh, the dyes in the river will decolorize by the microleca, and then the river is become clean, and the microleca still have there. So we can use like a filtration, or the other is we uh, pump the river into the tank containing the microleca, and then directly put the water into the river again, like this. So are this disturbing the balance of the ecosystem? The mycorrhiza is a fungi, non-toxic fungi. Yeah. And if there are so many nutrients for their growth, they will no grow dramatically. So this is not no problem for the environment. Or the other application is using the enzyme. Yeah, enzyme immobilized in leca, but in my experience, enzyme is easily broken by the temperature or by the toxic compound from the river. Yeah. So most potential using the microleca for treated the wastewater, I think.
Yeah. Can we know what ratio between microleca in wastewater? Let's say in 1 liter of wastewater, how many microleca do you use to this treatment?
Yeah, we already optimized using the responsive phase methodology. Yeah, using the composite central design, we obtained that 2 grams of microleca is suitable for 80 milliliter or 100 milliliter. 2 grams per 100 milliliter. So if 1 liter is around 20 grams, it's very small.
Yeah. Okay. Thank you again, Pak Dede. Next, which one? Okay. The orange colorant hardly decolorized using the microleca. It seems that there is another peak around 5.0. What do the peaks mean? It seems that the intensity decreased. So could you elaborate this, Pak Dede?
Okay. I will show the pic. Okay. So can I share screen? Okay. Okay. Do you mean this one? Do you mean this one? No?
Okay, maybe for the participant who asked this question,
This
please.
one, around 5, right?
Yeah, we guess so.
Yeah.
Yes, bro. Yeah.
This is the metabolite product. You can see that after before the acid orange are degraded, the peak is only acid orange right here, and after 72 hours, yeah, we collect all the peak, and this peak is one of these azo AOTP means acid orange uh, treated product, yeah, product after treated acid orange. So this compound is one of these product. Yeah. You can see that why the retention time of this peak is lower compared than the acid orange, because the most component is simple more simple compared than the azo that is here. So this is therefore the peak is only here. And then after that, the peak is higher, yeah, you too that. But after checking by the toxicity, it cannot be more toxic compared than only azo dye. No, acid orhan dye. As you can see here, oh, I didn't show the toxicity analysis here, but in the paper we measure the gain and toxicity of this all treatment, yeah, after 120 hour compared than a 0 hour. No toxic compared than only acid oil 7 like this. Yeah.
Okay, that is Arum, the answer. Hope it can answer your question.
Please
Next, we move to another question.
Number school.
Which one? Uh, ini related to the fungi application for the decolorization. Do we need to extract, or the grounded fungi could also work well to decolorize? Yeah, fungi in the land maybe.
Oh, okay. So actually, we call the indigenous microbe also can help the decolorization or degradation, but the indigenous fungi or indigenous microbials has the limitation, yeah, because of sometimes they didn't produce, for example, the lacase enzyme, but the lacase enzyme is very important to decolorize the textile dyes. So we need to we call, yeah, in the bioremediation terminology, we will do biostimulation. Yeah, biostimulation, we need to add the nutrient into the site, or bioaugmentation. Bioaugmentation means we need to add microbes into the area.
All right. Thank you, Pak Dede. We move to the next question from Ifah Munifah. Is it possible to use the mineral salid repeatedly?
Yeah, it's already answered.
It's already,
right?
yeah. Okay. Okay, the last two question maybe, Pak Dede. Dr. Dede, is it possible to use mycolica to reduce heavy metal pollution such as PB, HG, and others, maybe via absorption or kind of like that?
Yeah. Like has been reported to be used as a heavy metal absorption. Yeah, there's why we use mycolica for this. We use leca for as a matrix of the immobilization, because we hope that the leca can help absorption, yeah, instead of the biodegradation by the microbe. So it's possible, but research on bioabsorption, heavy metal bioabsorption by the fungi is already conducted by several researchers. But yeah, the mechanism, yeah, is different. Absorption is only absorb the heavy metal into the matrix. By using the microbes or fungi, the microbes or fungi can influence to change the valency of the heavy metal. For example, mercury, Hg, is a toxic when it exists as a organic metal, but it cannot be toxic into the ion condition. So how the microbe influence the change of the valency of the heavy metal. This is very important. Yeah. For example, arsenic is toxic in some condition, but not non-toxic in other condition. For example, PB is toxic for in ionic condition or in the metal conditions or like this. So microbes will influence into the change of the valency of the heavy metal.
All right. So basically, Mycoleka can use as a sorbent absorption media for heavy metal, but Pak Dede don't conduct this
Yeah.
experiment. Yeah. Okay.
Yes.
Last question in the AHA slide. There must be huge amount needs of the fungi for the decolorization, as Indonesia has large water contamination. Do you think we can fulfill these needs by harvesting the fungi, or do have to synthesize?
Yes. Okay. Yeah. One method is by immobilization, yeah, by immobilization or by making a microbial composite engineering. So after the mycelia fungi immobilized in matrix, then they will be there forever, yeah, as long as we can maintain the nutrient and the situation of for their growth, yeah. So the fungi can grows, yeah. We don't need to harvest the fungi. We only need grow them into their suitable media. Therefore, the putting their mycelia into the matrix is important. After we put in the matrix, then uh, whenever the mineral or the media coming or the dyes coming into this composite microbe, the fungi can go there. Yeah, this is the method for the application. We don't need
Okay.
to harvest.
Yeah. Okay, thank you, Pak Dede. We have one last question that has been given by participant in the chat room. Probably she or he forget to put the question in the slide. It is from Pepe. Pepe, would you like to ask Pak Dede directly, maybe, Pepe? Okay, otherwise I will read it for you. Yeah. Is there any field application have been done for the wastewater treatment using white rot fungi? How's the results and how many days used to purify the wastewater?
Yes, we already conducted in Pekalongan in one of the batik home industry, and also we already conducted in PT Jababeka Jakarta. PT Jababeka Jakarta is a batik wastewater treatment. No, textile wastewater treatment. Yeah, a very complex component in the wastewater. So, yeah, how many days? For example, if you see my presentation, around three weeks, yeah, uh, can be treated. But this is using the continuous system, using the continuous. I mean, after the treated, then after the 3 weeks, it can be directly normal, can uh, decolorize the textile dyes. Yeah, there, there is.
Okay, thank you, Pak Pepe. But I have one last question for by myself. Yeah, I just wondering. Yeah, is there any explanation about how fungi can decolorize the dye? What the mechanism to change the color, or mechanism for break the bond in the azo dyes? Is there any explanation so far?
Yeah, actually by using the breaking down the double bond in the uh, uh, benzene ring. For example, I will show you.
Yeah.
So
So basically, fungi can produce substance that can break the bond.
yes,
Isn't it?
enzyme. The
Oh,
enzyme. For
okay.
example, the
Okay.
lactase.
Yeah,
Yeah,
yeah, yeah.
yeah. The most for example, this is the conjugate link. Yeah. Uh, the color become dark, and after we broken this azo dyes, then the color become colorless because the conjugate double bond is broken by the lactase enzyme. So fungi produce the lactase enzyme, and then the lactase enzyme degraded this component.
All right. I see. I see. Yeah, yeah, yeah. Okay, thank you so much, Pak Dede, for the clear explanation to many questions that has been given to you. We are, yeah, we feel very clear about biomaterial, especially about mycoleka and so many work that you have been explained for us. And we move to the next session, maybe. Yeah, all right. We are at the end of the first session, and we really praise the knowledge that has been given by Dr. Dede from Brain. And moreover, on behalf of Department of Chemistry Education, we would like to show our gratitude to Pak Dede for your lecture, for your explanation, and for your insightful knowledge for us. For that, please receive the certificate of appreciation as the invited speaker at the virtual summer school 2022 Green Sustainable Chemistry for a Better Future. Once again, for all attendees, please give praise and appreciation for Pak Dede. Thank you so much, Pak Dede.
Thank you so much for this certificate and also for the committee. Yeah.
All
Thank you,
right.
Pamora.
Here's with us Bu Fitri as chief of virtual summer school. Maybe Bu Fitri, you have something to speak to Pak Dede, please.
All right. Thank you, Mora. Very good morning, Pak Dede. Yeah, it's
Yeah,
more
thank you,
than
Bu.
glad to enable to seeing you again. So on behalf of chemistry department, I would like to extend our thanks for your great acceptance and also your great effort. Yeah, to serve as the invited speaker in this
Yes.
program. Yeah, I do agree with the statement that just Mura mentions. So the materials that just delivered by Dr. Dede, in my opinion, was strongly related, yeah, to the topic of our Pitwa Summer School. Especially when we are questioning about the green chemistry for sustainable development. How to say, many innovation that just mentioned by Pa Dede, how to utilize various type of biomaterial, yeah, that abundantly
Yes.
available surrounding us. It could be a great hint how we engage, how we contribute to the sustainable development goal. And hopefully, I guess by extending applications and also development, yeah, of new material based on biomaterials. In the future, I guess, yeah, we can contribute significantly, yeah, to solve many problems that been
Yes.
occur in our environment, yeah, due to the excessive use of this synthetic material. So, Pak Dede, thank you so much for your great lectures, and
Yeah.
I do believe all participants also enjoy, yeah. We can see from so many questions just raised by the participants. And yeah, I also believe that your lectures can also increase, how to say, our awareness, yeah, how we can take into account the great potential in our environment. And then, yeah, we can do some innovations later. Thank you,
Yeah,
Pak
thank
Dede, and
you.
hopefully we can see you again in other
Yeah.
occasions. And yeah, thank
Yeah.
you so much.
Thank you so much, Bu Fitri, and we hope we can collaborate for next
Oh,
future. Okay,
very
thank you.
pleased, Pak Dede. Thank you so much.
Thank you so
Have a good
much.
day, Pak Dede. Yeah.
Yeah, thank you.
All right, thank you, Bu Fitri. Before we move to photo session. Maybe Pak Dede have any closing remarks for participants?
Okay, thank you. So I would like to again thanks to the committee, especially Bu Fitri, for inviting me to this lecture. Yeah, as mentioned by Bu Fitri, we have so many biomaterials around us, and we can utilize this as excellent material for a green and sustainable future for us. Yeah. So we invite all of the students and also lecturer here to collaborate with us, and you can access all the equipment in the in Brain for next collaboration. Yeah. Thank you very much again.
Okay, thank you so much once again to Pak Dede. And before Pak Dede leave us, please, we will have a photo session. So for all participants here, can you please to open your camera to document this memory, to document this event, and my fellow committee will help us to document this session. So please, participant, if you have any issue about connection, can you please to open your camera?
So just the additional information for Pak Dede. The participant who are attending this virtual summer school actually comes from many institutions. So in our record, around 42 participants come from abroad, yeah, from Philippines, Thailand, Malaysia, and also
nice.
Bangladesh, and then India, if I'm not mistaken, yeah, and also from Korea, from Japan, so and also from other national university in Indonesia. So yeah, it will be great, Pak Dede, in the future if they can, how to say, extend, yeah, the connections with you.
Okay, thank you very much. Yeah, very nice.
Okay, so Bu Vidya will help us to document this moment.
All right, everyone, please prepare your smile. We have 3 slides here. Okay, I will count to 3, 2, 1 for the first slide, smile. And then the second one. The last one. All right, that's all. Thank you to participants.
Okay. Thank you for all participants. And again, we share all thanks to Pak Dede for the lecture, and hopefully we can see you again in another occasion. Yeah. Thank you so much. Pak Dede can leave us if you have any other business.
Thank you
Thank you
very
so
much,
much, Pak
Pak Mas
Dede.
Mola, ya, Mbak Bu Fitri dan Bu Vidya. Thank you. See you again.
All right, everybody, all participants, we now move to usual session, which is quiz. Please prepare your quiz, and committee will share to you the link for the quiz in the slide. Yeah. So please. Okay. In the chat room. Yeah. The link for the quiz has been given to you in the chat room, slide.com C7KZZ. So please, everybody participant can enter the room, and yeah, we can start immediately the quiz. Okay, while we are waiting, because this is a little issues about sound. All right, I've seen 36 participants has already joined us. Okay. So all attendees, please join the slide in this link so we can start our quiz immediately. And this quiz will cover all the material that has been given by Pak Dede. So if you are stay tuned with us from the first beginning until the last time, I think you can answer all the question and get the pole position, yeah, in this quiz. Yeah, for your information, yang yeah, Priya has win 2 times in this quiz session. So could she be a champion again in this quiz session? Yeah, we will see. Yeah. Okay, 34 player have joined us right now. 35, still counting. Okay, I think has been stagnant, right? Yeah, all the participants. So we can start our quiz, and the quiz will be started in 3, 2, 1, and let's go. Here will be 8 questions. The first one: which one of component has the highest percentage in lignocellulose? Hemicellulose, cellulose, lignin, or tannin? Cellulose, yeah, like the name. Okay, who's at the pole position right now? Aslan. Okay, next to the second question. Which one is not major area in bionic prospecting? Additive, sensor technologies, architecture, or design? The answer is food additive. Yeah. Okay. Who's the leader right now? Mansoor. Congratulations. Let's move to the third question. Which one is the physical method to treat wastewater? Okay. It's already been explained by Pak Dede. Oxidation, ozonation, flocculation, or enzyme treatment. It's clear. Yeah, flocculation. Yeah, next. Okay, Mansur still lead the leaderboard. Congratulations. Next, number 4. Conventional and advanced technique used for wastewater treatment, except chemical methods, biological method, physical method, or the last one, computational method. I think all participants know what is the answer. Conventional. Okay, that's good. Yeah, that's good. Let's move to another question. Mansur still lead the leaderboard. Simple physicochemical process is the advantage of what technique? Flocculation, membrane filtration, biological filtration, or chemical method? Simple physicochemical process. That's the clear clue, the hint. Okay, the answer is flocculation. Absolutely. Yeah. Who's the leader right now? Oh, it's been changed to Anissa and Ivani. Yeah. Next, number 6. These followings are the advantages of using protein biomaterial, except reusability, operational stability, storage stability, or last one, heat stability. Protein biomaterials advantages. I think it's already mentioned in the Pak Dede slide. Heat stability. Yeah. The last 2 questions, I think. Anissa Maimuna, congratulations, you lead the line. Yeah. Thermal analysis can be characterized using this instrument. Example, it's very common. TGA, STEM, DTG, DSC, especially for physical chemistry student. You must know the answer of this exam, yeah. Okay, of course, Sam, yeah. Okay, the last question. Okay, we are Anissa still lead the line, followed by Ivani and FZ. And the very last question, Lekes can be used for the following application. Exam. Yeah, this is the last part of Pak Dede explanation. Antimicrobial agent, anti-diabetic agent, hair dyes, or personal care. The answer is anti-diabetic agent. And who's the winner of this session? Anissa Maimuna. Congratulations. Why the emoticon is very sad. Congratulations once again for Anissa Maimuna, the winner of this quiz session. All right, thank you committee for provide us the quiz, and yeah, we have arrived in the end of the session 1. Yeah, wait. And yeah, I will remind you that we still have the afternoon session with Professor Sung Hyun Lee, and the topic is wastewater treatment, yeah, innovation in wastewater treatment, and will be start from 1 p.m. Yeah, so you can have a little rest, have a lunch, and before we stop the conversation, maybe Bu Vidya, you have any announcement maybe for the tomorrow session, because there will be a lot of change right for tomorrow session. So please, Ibu Vidya, give us an announcement.
All right. Thank you, Kamora. For tomorrow, actually tomorrow will be the last lecture sessions. Because on Saturday we will have a please, okay. On Saturday we will have free time, so and then we continue our session on Sunday. So for tomorrow, okay. So for tomorrow, we will start at 8 a.m., but the Zoom will be open since from 7:30. So you can join the Zoom meeting on 7:30, but we will begin the lecture at 8 a.m. So the first lecture will be delivered by Professor Paulok Shaw from Malaysia. So at the first, he was scheduled to deliver the talk on Saturday, but unfortunately, due to some circumstances, that Professor Paulo Shaw can deliver his talk on Friday morning. So tomorrow we will start at 8 a.m. It will be delivered by Professor Paulo Shaw. And then we will have the second speaker at around 9:30 a.m. There will be Professor Rizalman from Malaysia. So for tomorrow's morning session, we will be having two speakers. First is Professor Faulok Show. We will start at 8 a.m. And then the second is Professor Rizalman. We will start at 9:30 a.m. So make sure that you don't miss the tomorrow lectures, yeah, because in the morning we have two speakers. Professor Paulo Shaw will be talking about renewable and clean energy, while Professor Rizaman, I think it's the same, yeah? Okay, Professor Rizaman also will be talking about the energy. And in the afternoon session, so for tomorrow's afternoon session, we will start at 1:30 p.m. 1:30 in the afternoon. Yeah, because for Muslim, tomorrow we will have a Friday prayer. Yeah, so we will start at 1:30 p.m. for tomorrow's afternoon. So make sure that for tomorrow you don't you can join us on time, because we will start at 8 a.m. and the Zoom meeting will be open at 7 will be open from 7:30 a.m. And also don't forget that tonight is the deadline for the poster. So make sure that you already that your group already created your own poster, and then you can submit it through the Padlet. The link for Padlet is already shared in the chat. So and after and also there will be assessment. Yeah, after all the posters been submitted to Padlet, there will be assessment. I think that's the announcement.
Thank you so much, Bu Vidya, for the announcement. So don't forget, tomorrow the session will be start at 8 a.m. with 4 speaker. Yeah, the last day for topic. Yeah. All right, everybody. I think you are all hungry, so we will stop until this. So see you again at 1 p.m. We will have a speaker from Korea, Prof. Suyun Lee, and will be delivered us topic about innovation in wastewater technology. Don't miss it. So see you at 1 p.m. Bye-bye. Oh yeah, don't forget to fill up the attendance link. Yeah, the attendance link has been shared to you in the chat room. Yeah, don't forget to fill it.
Located in the green premise of Universitas Pendidikan Indonesia, Bandung, West Java, the Department of Chemistry Education is one of the oldest departments in UPI, initially established as the Natural Science Program in 1954. The Department of Chemistry Education occupies Japanese architecture JICA building, whose name depicts strong collaboration with the Japan International Cooperation Agency. We provide an academic atmosphere for a satisfying experience in studying chemistry and chemistry education. Aligned with the UPI's vision, the Department of Chemistry Education under the Faculty of Mathematics and Natural Sciences Education is determined to be a leading and outstanding department in chemistry and chemistry education. The Department of Chemistry Education strives to prepare professional chemistry teachers and chemists who can compete globally, also establish and strengthen national, regional, and international networks. These missions are supported by 7 professors and 40 academics, consisting of 24 lecturers holding a doctoral degree, and 16 lecturers are prospective doctoral candidates. The Department of Chemistry Education offers 4 study programs, including Bachelor of Chemistry, Bachelor of Chemistry Education, Master of Chemistry, and Master of Chemistry Education programs. 3 study programs have been accredited A by the National Accreditation Council for Higher Education. This rank indicates high qualification and recognition of study programs in fulfilling the national education standards. For Master of Chemistry program as a new study program has met the minimum requirements for accreditation. The 4 study programs are now undergoing the accreditation process by the International Accreditation Board. With the existence of educational and non-educational programs, the Department of Chemistry Education implements the cross-fertilization principle to build synergy between the 2 programs and strengthen the 3 pillars of higher education that consist of education, research, and community service. The curriculum structure in the Department of Chemistry Education is designed to prepare students either for professional work or for graduate studies. At the bachelor level, students will be studying fundamentals of chemistry, analytical chemistry, physical chemistry, inorganic chemistry, organic chemistry, biochemistry, applied chemistry, lab experiments, and additional course of chemistry education for students of the educational program. While at the master level, students will be exposed to a more comprehensive knowledge and research of chemistry, chemistry education, and their applications. The Department of Chemistry Education is supported by 4 expertise or research groups in the chemistry program and 7 in the chemistry education program to excel at the 3 pillars of higher education. The chemistry program specializes in material chemistry, environmental chemistry, biological chemistry, and food chemistry. Material chemistry group focuses on the transformation of biomaterials or waste materials into nanomaterials, porous materials, and hybrid materials that can be used as catalysts, corrosion inhibitors, adsorbents, and membranes. Environmental Chemistry Group conducts research on bionutrients, biohydrogels, membranes, thin films, bionanomaterials, nanocomposites, and modeling studies. Food Chemistry Group manages research on food fortification, food preservation, functional food, food safety and security, aiming to produce high-quality food. Biological Chemistry Group researches exploration and modification of natural products from Indonesia that can be useful for application in environment, energy, health, and food. The expertise group in the chemistry education program consists of chemistry literacy for education for sustainable development or ESD and teacher professional development, high-order thinking skills or HOTS in chemistry education, chemistry in context or chemicon, read, answer, discuss, explain, create, curriculum, problem-based learning, intertextuality in chemistry learning, innovative assessment in chemistry, and digital media for learning chemistry. The research development at the Department of Chemistry Education is facilitated through many research grants worth billions of rupiah each year, local, national, and international fellowships, and strengthened through partnerships with government, industry, research institutes, and other higher education institutions. The Department of Chemistry Education provides students with facilities to support all student activities from learning to student well-being. The teaching and learning process are conducted in well-equipped classrooms and laboratories. The Department of Chemistry Education is facilitated with the smart class equipped with a smart board and sophisticated audio-visual media to support digital-based learning and interactive and collaborative teaching. To carry out independent learning and e-learning, the Department of Chemistry Education also uses the Learning Management System or LMS established by the university, such as Integrated Online Learning System or SPOT and Online Learning System or SPADA. These platforms enable an easy and more flexible learning process. The Department of Chemistry Education has 9 laboratories equipped with standard and advanced instruments to support practicum and research, including Laboratory of Basic Chemistry and Analytical Chemistry, Laboratory of Physical Chemistry and Inorganic Chemistry, Laboratory of Organic Chemistry and Biochemistry, Laboratory of Analytical Instruments, Laboratory of Teaching and Learning to support chemistry learning simulation and research in the chemistry education program, and 4 research laboratories. The laboratories are accessible to all staff and students of UPI as well as external researchers. To promote safe practice, the labs are equipped with safety equipment, standard operating procedures or SOPs, and personal protective equipment or PPE. In addition to standard laboratory equipment, students and staff can access various instruments including UV-vis spectrophotometer, Fourier transform infrared or FTIR spectrometer, high-performance liquid chromatography or HPLC, gas chromatography mass spectrometry or GCMS, atomic absorption spectrophotometer or AAS, liquid chromatography with tandem mass spectrometry or LCMS-MS, thermogravimetric analyzer, and computational modeling facility. Other supporting facilities served for students to have a convenient learning experience include library, language center, art and sports facilities, University Training Center or Isla Resort, Center for Counseling and Guidance, Career Development Center, Polyclinic, Mosque, Student Center, and Student Dormitory. The Department of Chemistry Education has established collaborations with national and international institutions, including research collaboration, joint publications, visiting lecturers or researchers, students' mobility, internship programs, staff development, and community services. To promote international engagement, the Department of Chemistry Education also holds an international summer school annually, where students from all over the world can connect and meet experts. We also allows students to absorb knowledge and experience a new learning atmosphere across countries through a student mobility program. The successful outcomes of learning and research activities at the Department of Chemistry Education are depicted through students' achievements. Becoming medalist in Chemistry National Olympiad and Micro-Teaching Competition, receiving grants for student creativity program, publishing papers in reputable international journals, and receiving intellectual property rights for the research work are to name a few. To encourage vibrant life and make the most of university life, students are offered plenty of opportunities to connect with other students and improve soft skills. These include community service programs, workshops, sports, art, and extracurricular activities. The student body routinely organizes numerous events involving societies and schools such as Dis Natalis, Chemistry Expo, seminars, and charities. To date, graduates of the Department of Chemistry Education have contributed to the development of education and research in national and global communities. Welcome to Department of Chemistry Education, Universitas Pendidikan Indonesia. Department of Chemistry Education, Universitas Pendidikan Indonesia, leading and outstanding. Located in the green premise of Universitas Pendidikan Indonesia, Bandung, West Java, the Department of Chemistry Education is one of the oldest departments in UPI, initially established as the Natural Science Program in 1954. The Department of Chemistry Education occupies Japanese architecture JICA building, whose name depicts strong collaboration with the Japan International Cooperation Agency. We provide an academic atmosphere for a satisfying experience in studying chemistry and chemistry education. Aligned with the UPI's vision, the Department of Chemistry Education under the Faculty of Mathematics and Natural Sciences Education is determined to be a leading and outstanding department in chemistry and chemistry education. The Department of Chemistry Education strives to prepare professional chemistry teachers and chemists who can compete globally, also establish and strengthen national, regional, and international networks. These missions are supported by 7 professors and 40 academics, consisting of 24 lecturers holding a doctoral degree, and 16 lecturers are prospective doctoral candidates. The Department of Chemistry Education offers 4 study programs, including Bachelor of Chemistry, Bachelor of Chemistry Education, Master of Chemistry, and Master of Chemistry Education programs. 3 study programs have been accredited A by the National Accreditation Council for Higher Education. This rank indicates high qualification and recognition of study programs in fulfilling the national education standards. For Master of Chemistry program as a new study program has met the minimum requirements for accreditation. The 4 study programs are now undergoing the accreditation process by the International Accreditation Board. With the existence of educational and non-educational programs, the Department of Chemistry Education implements the cross-fertilization principle to build synergy between the 2 programs and strengthen the 3 pillars of higher education that consist of education, research, and community service. The curriculum structure in the Department of Chemistry Education is designed to prepare students either for professional work or for graduate studies. At the bachelor level, students will be studying fundamentals of chemistry, analytical chemistry, physical chemistry, inorganic chemistry, organic chemistry, biochemistry, applied chemistry, lab experiments, and additional course of chemistry education for students of the educational program. While at the master level, students will be exposed to a more comprehensive knowledge and research of chemistry, chemistry education, and their applications. The Department of Chemistry Education is supported by 4 expertise or research groups in the chemistry program and 7 in the chemistry education program to excel at the 3 pillars of higher education. The chemistry program specializes in material chemistry, environmental chemistry, biological chemistry, and food chemistry. Material chemistry group focuses on the transformation of biomaterials or waste materials into nanomaterials, porous materials, and hybrid materials that can be used as catalysts, corrosion inhibitors, as solvents, and membranes. Environmental Chemistry Group conducts research on bionutrients, biohydrogels, membranes, thin films, bionanomaterials, nanocomposites, and modeling studies. Food Chemistry Group manages research on food fortification, food preservation, functional food, food safety and security, aiming to produce high-quality food. Biological Chemistry Group researches exploration and modification of natural products from Indonesia that can be useful for application in environment, energy, health, and food. The expertise groups in the chemistry education program consist of chemistry literacy for education for sustainable development or ESD and teacher professional development, high-order thinking skills or HOTS in chemistry education, chemistry in context or ChemiCon, read, answer, discuss, explain, create curriculum, problem-based learning, intertextuality in chemistry learning, innovative assessment in chemistry, and digital media for learning chemistry. The research development at the Department of Chemistry Education is facilitated through many research grants worth billions of rupiah each year, local, national, and international fellowships, and strengthened through partnerships with government, industry, research institutes, and other higher education institutions. The Department of Chemistry Education provides students with facilities to support all student activities from learning to student well-being. The teaching and learning process are conducted in well-equipped classrooms and laboratories. The Department of Chemistry Education is facilitated with the smart class equipped with a smart board and sophisticated audio-visual media to support digital-based learning and interactive and collaborative teaching. To carry out independent learning and e-learning, the Department of Chemistry Education also uses the Learning Management System or LMS established by the university, such as Integrated Online Learning System or SPOT and Online Learning System or SPADA. These platforms enable an easy and more flexible learning process. The Department of Chemistry Education has 9 laboratories equipped with standard and advanced instruments to support practicum and research, including Laboratory of Basic Chemistry and Analytical Chemistry, Laboratory of Physical Chemistry and Inorganic Chemistry, Laboratory of Organic Chemistry and Biochemistry, Laboratory of Analytical Instruments, Laboratory of Teaching and Learning to support chemistry learning simulation and research in the chemistry education program, and 4 research laboratories. The laboratories are accessible to all staff and students of UPI as well as external researchers. To promote safe practice, the labs are equipped with safety equipment, standard operating procedures or SOPs, and personal protective equipment or PPE. In addition to standard laboratory equipment, students and staff can access various instruments including UV-vis spectrophotometer, Fourier transform infrared or FTIR spectrometer, high performance liquid chromatography or HPLC, gas chromatography mass spectrometry or GCMS, atomic absorption spectrophotometer or AAS, liquid chromatography with tandem mass spectrometry or LCMS-MS, thermogravimetric analyzer, and computational modeling facility. Other supporting facilities served for students to have a convenient learning experience include library, language center, art and sports facilities, university training center or Isla Resort, center for counseling and guidance, career development center, polyclinic, mosque, student center, and student dormitory. The Department of Chemistry Education has established collaborations with national and international institutions, including research collaboration, joint publications, visiting lecturers or researchers, students' mobility, internship programs, staff development, and community services. To promote international engagement, the Department of Chemistry Education also holds an international summer school annually, where students from all over the world can connect and meet experts. We also allow students to absorb knowledge and experience a new learning atmosphere across countries through a student mobility program. The successful outcomes of learning and research activities at the Department of Chemistry Education are depicted through students' achievements. Becoming medalist in Chemistry National Olympiad and Micro-Teaching Competition, receiving grants for Student Creativity Program, publishing papers in reputable international journals, and receiving intellectual property rights for the research work are to name a few. To encourage vibrant life and make the most of university life, students are offered plenty of opportunities to connect with other students and improve soft skills. These include community service programs, workshops, sports, art, and extracurricular activities. The student body routinely organizes numerous events involving societies and schools such as Dis Natalis, Chemistry Expo, seminars, and charities. To date, graduates of the Department of Chemistry Education have contributed to the development of education and research in national and global communities. Welcome to Department of Chemistry Education, Universitas Pendidikan Indonesia. Department of Chemistry Education, Universitas Pendidikan Indonesia, leading and outstanding. 2024. The Department of Chemistry Education occupies Japanese architecture JICA building, whose name depicts strong collaboration with the Japan International Cooperation Agency. We provide an academic atmosphere for a satisfying experience in studying chemistry and chemistry education. Aligned with the UPI's vision, the Department of Chemistry Education under the Faculty of Mathematics and Natural Sciences Education is determined to be a leading and outstanding department in chemistry and chemistry education. The Department of Chemistry Education strives to prepare professional chemistry teachers and chemists who can compete globally, also establish and strengthen national, regional, and international networks. These missions are supported by 7 professors and 40 academics, consisting of 24 lecturers holding a doctoral degree and 16 lecturers are prospective doctoral candidates. The Department of Chemistry Education offers 4 study programs, including Bachelor of Chemistry, Bachelor of Chemistry Education, Master of Chemistry, and Master of Chemistry Education programs. 3 study programs have been accredited A by the National Accreditation Council for Higher Education. This rank indicates high qualification and recognition of study programs in fulfilling the national education standards. For Master of Chemistry program as a new study program has met the minimum requirements for accreditation. The 4 study programs are now undergoing the accreditation process by the International Accreditation Board. With the existence of educational and non-educational programs, the Department of Chemistry Education implements the cross-fertilization principle to build synergy between the 2 programs and strengthen the 3 pillars of higher education that consist of education, research, and community service. The curriculum structure in the Department of Chemistry Education is designed to prepare students either for professional work or for graduate studies. At the bachelor level, students will be studying fundamentals of chemistry, analytical chemistry, physical chemistry, inorganic chemistry, organic chemistry, biochemistry, applied chemistry, lab experiments, and additional course of chemistry education for students of the educational program. While at the master level, students will be exposed to a more comprehensive knowledge and research of chemistry, chemistry education, and their applications. The Department of Chemistry Education is supported by 4 expertise or research groups in the chemistry program and 7 in the chemistry education program to excel at the 3 pillars of higher education. The chemistry program specializes in material chemistry, environmental chemistry, biological chemistry, and food chemistry. Material chemistry group focuses on the transformation of biomaterials or waste materials into nanomaterials, porous materials, and hybrid materials that can be used as catalysts, corrosion inhibitors, adsorbents, and membranes. Environmental Chemistry Group conducts research on bionutrients, biohydrogels, membranes, thin films, bionanomaterials, nanocomposites, and modeling studies. Food Chemistry Group manages research on food fortification, food preservation, functional food, food safety and security, aiming to produce high-quality food. Biological Chemistry Group researches exploration and modification of natural products from Indonesia that can be useful for application in environment, energy, health, and food. The expertise group in the chemistry education program consists of chemistry literacy for education for sustainable development or ESD and teacher professional development, high-order thinking skills or HOTS in chemistry education, chemistry in context or ChemiCon, read, answer, discuss, explain, create, curriculum, problem-based learning, intertextuality in chemistry learning, innovative assessment in chemistry, and digital media for learning chemistry. The research development at the Department of Chemistry Education is facilitated through many research grants worth billions of rupiah each year, local, national, and international fellowships, and strengthened through partnerships with government, industry, research institutes, and other higher education institutions. The Department of Chemistry Education provides students with facilities to support all student activities from learning to student well-being. The teaching and learning process are conducted in well-equipped classrooms and laboratories. The Department of Chemistry Education is facilitated with the smart class equipped with a smart board and sophisticated audio-visual media to support digital-based learning and interactive and collaborative teaching. To carry out independent learning and e-learning, the Department of Chemistry Education also uses the Learning Management System, or LMS, established by the university, such as Integrated Online Learning System, or SPOT, and Online Learning System, or SPADA. These platforms enable an easy and more flexible learning process. The Department of Chemistry Education has 9 laboratories equipped with standard and advanced instruments to support practicum and research, including Laboratory of Basic Chemistry and Analytical Chemistry, Laboratory of Physical Chemistry and Inorganic Chemistry, Laboratory of Organic Chemistry and Biochemistry, Laboratory of Analytical Instruments, Laboratory of Teaching and Learning to support chemistry learning simulation and research in the chemistry education program, and 4 research laboratories. The laboratories are accessible to all staff and students of UPI as well as external researchers. To promote safe practice.
Good afternoon, Prof. Lee.
Okay, thank you. Thank you for inviting me. Can you hear me?
Yes. All's clear. And
Okay.
we will start in 1 p.m., Prof. So we're waiting for a bit longer.
Okay,
Thank you.
thank you.
The labs are equipped with safety equipment, standard operating procedures or SOPs, and personal protective equipment or PPE. In addition to standard laboratory equipment, students and staff can access various instruments including UV-Vis spectrophotometer, Fourier transform infrared or FTIR spectrometer, high performance liquid chromatography or HPLC, gas chromatography mass spectrometry or GCMS, atomic absorption spectrophotometer or AAS, liquid chromatography with tandem mass spectrometry or LCMS-MS, thermogravimetric analyzer, and computational modeling facility. Other supporting facilities served for students to have a convenient learning experience include library, language center, art and sports facilities, university training center or Isla Resort, Center for Counseling and Guidance, Career Development Center, Polyclinic, mosque, student center, and student dormitory. The Department of Chemistry Education has established collaborations with national and international institutions, including research collaboration, joint publications, visiting lecturers or researchers, students' mobility, internship programs, staff development, and community services. To promote international engagement, the Department of Chemistry Education also holds an international summer school annually, where students from all over the world can connect and meet experts. We also allow students to absorb knowledge and experience a new learning atmosphere across countries through a student mobility program. The successful outcomes of learning and research activities at the Department of Chemistry Education are depicted through students' achievements. Becoming medalist in Chemistry National Olympiad and micro-teaching competition, receiving grants for student creativity program, publishing papers in reputable international journals, and receiving intellectual property rights for the research work are to name a few. To encourage vibrant life and make the most of university life, students are offered plenty of opportunities to connect with other students and improve soft skills. These include community service programs, workshops, sports, art, and extracurricular activities. The student body routinely organizes numerous events involving societies and schools such as Dis Natalis, Chemistry Expo, seminars, and charities. To date, graduates of the Department of Chemistry Education have contributed to the development of education and research in national and global communities. Welcome to Department of Chemistry Education, Universitas Pendidikan Indonesia. Department of Chemistry Education, Universitas Pendidikan Indonesia, leading and outstanding. Located in the green premise of Universitas Pendidikan Indonesia, Bandung, West Java, the Department of Chemistry Education is one of the oldest departments in UPI, initially established as the Natural Science Program in 1954. The Department of Chemistry Education occupies Japanese architecture JICA building, whose name depicts strong collaboration with the Japan International Cooperation Agency. We provide an academic atmosphere for a satisfying experience in studying chemistry and chemistry education. Aligned with the UPI's vision, the Department of Chemistry Education under the Faculty of Mathematics and Natural Sciences Education is determined to be a leading and outstanding department in chemistry and chemistry education. The Department of Chemistry Education strives to prepare professional chemistry teachers and chemists who can compete globally, also establish and strengthen national, regional, and international networks. These missions are supported by 7 professors and 40 candidates.
All right. Uh, Hello, Professor Lee. Shall we start the session right now? Okay.
Okay, great.
Yeah. Okay. Dear all participants, welcome back again to our virtual summer school day 4 in session 2. And as you know, the virtual summer school with theme Green Sustainable Chemistry for a Better Future. This event held by Department of Chemistry Education, Universitas Pendidikan Indonesia. I am Abraham Mora, you can call me Mora. And today, in this session, I will be your moderator again. And here with us has already joined Professor Sung Yoon Lee from Kyungpook National University, South Korea, that will give us a talk about innovation in wastewater treatment. This topic basically is related to the SDG number 6, which is clean water and sanitation. Following that, yeah, for your information, all the recorded version of this virtual summer school will be published in our YouTube channel, Departemen Pendidikan Kimia UPI. So don't miss to visit our YouTube channel, put your comment, like, and subscribe. And before we start our session with Professor Sung Yoon Lee, with innovation in wastewater treatment, we from committee have a glimpse video about wastewater treatment. So please enjoy.
Cities and towns have sewerage systems in place to carry the wastewater from our homes, hotels, factories, and other establishments to a sewerage treatment plant where the wastewater is treated. It is then deemed safe to release into other water sources. The treatment of wastewater goes through different stages. Wastewater is treated to remove the physical, chemical, and biological contaminants present in it through various processes. The pre-treatment process involves the sewage being sent through grids or vertical bars that can remove large solid substances like metal cans, paper, and plastic materials. In primary treatment process, the sewage flows through the grit chamber very slowly, so that the sand, pebbles, and soil settle down at the bottom. The sewage flows into the settling tank or sedimentation tank, where the solid wastes like feces are allowed to settle down. Wastes such as soaps, oils, and grease rise to the top of the wastewater. The waste material that settles down at the bottom is called the sludge, and the floatable material is called the scum. Scum is then removed using a skimmer, and sludge is removed with a scraper every few days. The water that is left out is called the clarified water. In the secondary treatment process, biological or organic wastes are removed. It is a biological process. This is done by transferring the clarified water into an aeration tank, where air blowers bubble air, which helps the aerobic bacteria to grow and feed on the organic contaminants such as food waste, feces, and other organisms. The mixture then flows from the aeration tank into the clarifier, where activated sludge settles out by gravity. The activated sludge so produced, along with the sludge produced in the primary process, is transferred to the digester, where it is decomposed by anaerobic bacteria. Biogas is produced in this process, which can be used as fuel or can be used to produce electricity. The activated sludge is then left in the sand drying beds. Some quantity of water in the activated sludge evaporates, and the remaining quantity drains through the sand beds. This leaves behind the remaining dried sludge, which can be used for making fertilizers or compost. In the tertiary treatment process, the leftover wastewater is treated with chlorine to remove the phosphorus compounds, nitrogen compounds, and bacteria. It is a chemical process. Chlorine tablets are added to kill the germs. This process is called chlorination. Then this water is let off into the water bodies.
All right, that is a short video that give us an overview for what Professor Lee will deliver for us about water treatment. And before we jump into the main season, I would like to read the curriculum vitae of Professor Lee first. All right, Professor Lee is currently a professor at Kyungpook National University, South Korea. He is focusing his research on the field of membrane forward osmosis as well as water treatment. He finished his bachelor degree in environmental engineering at Jeju University and continue his master and a PhD program in the environmental engineering Gwangju Institute of Science and Technology, South Korea. Next, as we can see here that there are so many publications that has been published by him. For example, I read for you about performance comparison of spiral wound and plate and frame forward osmosis membrane, and so many on that yeah, can tell us about his expertise in the water treatment. Next. Here are some patents that Professor Lee have. For example, column device for ion exchange resin for reducing regeneration chemical and operating method thereof, and so many on. And following by the Fellowship and Award in 2010 and 2008, postdoctoral research fellowship and best research award from GIST. So without any further ado, for Professor Lee, time is yours.
Okay, thank you for kind introduction. And I will open my presentation material first. Can you see clearly this material?
Yes, perfect, Professor Lee.
Okay. Start. Okay. Okay, first of all, I'd like to thank the committee of the virtual summer school for the kind invitation to give this lecture. And I'm really happy to meet you young students from Indonesia and other countries. And I hope this lecture is helpful for you. And I got the lecture topic as innovation in wastewater treatment, but I extended a little bit. I changed it a little bit to the innovation in water treatment, because water treatment and wastewater treatment, the principles are same. So I wanted to also talk about the desalination, which is a very important method to get the drinking water sources. So I extended this topic to the water treatment instead of wastewater treatment. Okay, I prepared several contents. First, I'd like to talk about environmental issues in the world and in Korea. And next, I will talk about the innovative technologies and what drives the invent of the new technologies. And then I will show you some of the example of the innovative water treatment technologies. And then finally, I'd like to share some topics. So I prepared some topics to share with you. So let me start with the environmental issues in the world and Korea. There is no doubt that the most important environmental issue in the world is climate change. Because of the climate change, we experience global warming, and according to the article, the global average temperature is the highest in past 4 years. And also we experience severe drought, and addition to the drought, we also sometimes have the very intensive heavy rain. In Korea, last week we had a very heavy rain last week, and it was the heaviest rain in 100 years. And it is expected that that kind of the sudden and heavy rain will increase in the future. So to protect the Earth and to reduce the climate change, UNFCCC agreed to have the more enforced commitment to reduce the emission of the CO2 gas. And the new climatic regime started from the last year. Although this effort still in Korea, we are emitting CO2 increasingly, and also in the world, the emission of CO2 is increases. And in 2016, in Korea, we made the greenhouse gas reduction roadmap, and also we revised this roadmap in 2018 to reduce the emission of the CO2 gas to cope with the Paris Agreement. And even though this effort, we are still increasing the emission of the CO2 gas, and global carbon dioxide emission is continued to rise. And interestingly, in European country, their carbon dioxide emission is decreased by 2.1% due to the renewable energy. This means that we have to change our energy source from the fossil fuel to the renewable energy to reduce the emission of the CO2 gas. And this kind of the climate change can be challenging for the environment, and the water shortage problem is very severe in Korea. But this kind of challenge can be the opportunity in the water business area. For example, because of the severe water shortage in the industrial complex in Daesan in Korea, our government decided to make the desalination, a huge desalination plant to supply sufficient water supply to the industry complex. So because of this kind of the challenge, maybe the water treatment industry can have some opportunity. So we have to develop new types of the better water treatment technology to reduce the energy consumption and to reduce the emission of the CO2. And also it can be conducted by improving the energy efficiency of the water treatment technologies. I'd like to talk about the water stress a little bit more. And this slide shows the prediction of the water stress in the world. The dark red represented the higher water stress, and the lighter color area means less water stress. And in Korea, we are having the medium to high water stress. And also according to this map, the Indonesia, some area has a severe water stress. Let's look at this slide. This slide shows the national water stress ranking. And as you can see here, most of the Middle Eastern countries belong to extremely high water stress country, and some of the European country also belong to the high water stress country. And in case of Korea, we ranked at the 53rd rank, and also Indonesia ranked to the 65th of the water stress country. So South Korea and Indonesia belong to the medium-high water stress country. And unfortunately, this kind of the water demand and water supply can have a more big difference in the near future. According to this article and this material, we can expect that around the 40% gap between the water demand and water supply in 2030. it means that we need to find a new method to have more water. And actually, there are several ways to have another water resources. And there are three main alternative water sources. First one is water reuse. The second one is collecting rainwater. And third one is desalination. In case of the water reuse, it can be conducted by upgrading existing the water treatment facility, and then we can have a stable water supply from the wastewater treatment plant. But one of the hurdle using for the water reuse is people's exception for the water reuse. For example, if we treat water from the wastewater treatment plant, and we can apply various fancy water treatment technologies, and we can remove all the pollutant from the wastewater. And if we measure the water quality using various instruments, maybe we can get very low or low pollutant in the water. But people will not drink, maybe reluctant to drink water from the wastewater because maybe there is a known component in the water. So that's why people don't want to drink water treated from the wastewater effluent. And the next one is collecting rainwater. It can be cost-effective solution to have the external water source, but sometimes it is not enough amount to use. So rainwater collection can be one option, but it's not a good way to supply sufficient water. However, in case of desalination, it is a good way to have stable and sufficient water because 97% of the Earth's water is seawater. So there are a lot of water, but it contains a high concentration of salt. So if we can remove the salt from the seawater efficiently, then we can have enough water. So desalination technology is a good way to have additional water source. I'd like to talk about a little bit specific environmental issue in Korea. Traditionally, the BOD or COD, biochemical oxygen demand or chemical oxygen demand, has been used to for the monitor of the removal efficiency of the wastewater. But however, the BOD or COD sometimes cannot measure non-degradable organic matter. So Korea Ministry of Environment revised the law to change the index for the organic carbon from COD to TOC. Therefore, there is increased need to measure TOC in wastewater treatment facility in Korea. So before we talk about the innovative water treatment technology, I'd like to talk about what is innovative technology and what drives invent of the innovative technology first. What is innovative technology? Can you imagine some keyword about the definition of the innovative technology? Okay. In 2016, about 6 years ago in Korea, historic event happened. Can you imagine what it is? It is relating to Go game match. It is not a normal Go game match. It was the AlphaGo versus the human. And it was a very big event in Korea. And also you may heard about this game. And this is a very historic event because after this event, ordinary people like me could know about the machine learning and deep learning. And that is a kind of the common keyword in research nowadays. So let's think about the innovative technology again. Can you imagine the keywords for the innovative technology? What it is? So maybe it should be cutting edge technology, right? And also some people can think about the combination with the technology with the ICT, information and communication technology. It should be maybe. And also maybe the keyword would be new technology. It should be new. How about the old one, older technology or modified technology? I think the modified technology also can be the innovative technology. So there are many keywords for the innovative technology, I think. And also it should be maybe automated technology. And also some people think about the green technology for the future. So before I talk about the innovative water technology, I'd like to define the definition. So this is my definition. I think the innovative technology is similar to the creative technology, and it is new or modified technology. And the important thing is it should be meet the needs. Then I think it is innovative technology. So to talk about the innovative technology, then I think we have to know about what is needs. Then we can think about the innovative technology again. What are the needs? Okay, what are the needs for the innovative environmental technology then? I think the first needs is technical needs. Technically, we want to remove some of the pollutant in the water, and existing or previous technology is difficult to remove that kind of pollutant. Then we have to find another technology to remove that kind of pollutant. So there will be technical needs for the innovative technology. And also I'm thinking about the economical. It should be economical. If the technology is too expensive, it is difficult to implement that technology. So we have to the economic needs for the innovative technology for the feasibility of the technology. And also, what else we need? And environmental. We have environmental needs, as we talked about at the first part of this lecture. There is climate change. So we want to stop the climate change. So that is kind of the environmental needs for the innovative water treatment technology. And also, I think social needs. There is a social needs. People don't want to pollute the environment, and also they want to drink very safe water. That is kind of social needs. And also the last one, what would be the last one? That is very important and very strong needs. That is law. We have to obey the law. If you break the law, then you have to pay fine and maybe you have to go to jail. So we have to obey the law. And so I think these 5 needs are important for the innovative water environmental technology. And some of the technology may have benefit in 1 or 2 of these needs, but at least I think it should, the technology should satisfy remaining the needs at least a certain level. And interestingly, I think all these are connected. So when we develop new technology or new water treatment technology, we have to think about all the needs. Let me show you some of the more detailed examples for the each needs. First, technical needs. I mentioned about the removal efficiency. So we have to remove the BOD, COD, and TOC, and we have to remove the pathogen from the water. And also sometimes we have to think about the removal of a boron. Have you heard about the boron? Boron is a kind of the key component included in the seawater. The concentration is around 5 milligrams per liter, but it is difficult to remove by the RO desalination plant. And also, if the product water has a high concentration of boron, maybe 1 milligram per liter, it can be harmful to the plants, like the agriculture purpose. The boron should be removed from the water. So the membrane should be developed to increase the removal efficiency of boron. So the removal efficiency is one of the technical needs, and also we want automated process. So we want to reduce our labor, and we don't want to do the repeated work, and there will be some regular maintenance. So this kind of repeated work we want to be automated. So we want automation for the as a technical needs. And sometimes we need real-time response from the system. Nowadays, real-time is very important. We are talking in real time like this. So real time is very important, and people want to check the system, whether it's working or not well. So maybe using the cell phone, we can check the working condition of the process. And also we want to check the pollutant emission concentration at the real time. So if we can measure the concentration of pollutant in real time, then we can have the quick response for the pollutant or some problems. So I think that these are the example of the technical needs. And also there are economical needs. As I mentioned, we should develop more efficient process, so then we can use the low energy. I think energy is synonym of the money, right? If we use a lot of money, energy, that means we use a lot of money for the process. So we want energy efficient process. And also sometimes we apply energy recovery device to get more energy and reduce input energy, especially for the desalination. And also if some material is expensive, then it is difficult to implement the technology. For example, at the first time, the membrane cost was very expensive, so it is difficult to apply the membrane to the desalination process. But after developing the membrane material, so we could reduce the price of the membrane, and also some other equipment like pump and other mechanical equipment should be cheap to operate the membrane process. So all equipment should be developed to reduce the cost. And also there is some operating cost. For example, the process can be cheap to install, but operating cost may be expensive. Maybe we have to give a lot of the expensive chemicals for the performance of the system, then that will be expensive. So we want to reduce the chemical use, and also to extend the lifetime of some process. Maybe we have to clean the process frequently. So then if we clean some process, then we can extend the lifetime of the whole process. Then we can reduce some operating cost. So there is some needs in the operating system relating to the economical needs. And also I mentioned about the environmental needs. So we have to think about the climate change, and also we think about the environmental impact. Relating to environmental impact, we sometimes conduct environmental impact assessment before we build some kind of building or facility in certain environment. And also to reduce environmental impact, we want to develop some kind of technology to reduce the production of the waste, and also we developed recovery technology from the waste that is relating to environmental impact. And also we want to use the less harmful chemical. For example, if some process needs strong acid and base for the cleaning, that would be harmful to the environment. So we want to find another types of the process sometimes, or we want to find another chemical to reduce harmful impact to environment. And I mentioned about the social needs. People want to use some process or product which is environmentally friendly. So people are interested in using biodegradable material to protect the environment. And we want to reduce the emission of the EDC compound for the ecosystem. And also aesthetic needs is very important. I mentioned about the wastewater reuse. And also there was an example in Korea too. For the desalination plant development in Korea, we build a big desalination, RO desalination pilot in Busan area. And after building the pilot plant, the researcher wanted to give the product of the desalinated water to the citizen in Busan city, but the citizen protested getting the water from desalinated water from that facility. One of the reasons was the location of the desalination plant was near to the nuclear power plant. Nuclear power plant maybe emit some kind of the pollutant which can emit some can I say the bio radiation, and they worried about the tritium H3 maybe included in the desalinated water. So this tritium can cause some cancer, so they don't want it to receive the water from the seawater pilot system. So our Korean government spent a lot of money to build that seawater desalination plant, but we cannot operate it for the citizen in the Busan because of the aesthetic needs of the people. So we have to think about aesthetic needs. And I already talked about automation before. And the final one is a law. Of course, we have to remove some pollutant before we emit the water, wastewater to the river or lake. And also, and there is, I talked about the Paris Agreement. Many countries have to reduce the emit of the CO2 emission. That is kind of the international law. So we have to follow the law. So that's why we have to invent new innovative technology. And also, that is a driver for the invention of the new technology. And also, law can be hurdle for some technology because law should permit using that kind of the new technology. For example, if you develop, let's assume that you developed a very good water treatment technology which can remove all the pollutant from the water with very low energy consumption. Then do you think you can implement that technology immediately? No, because of the law. Law wrote that what kind of the water treatment process can be used for the making drinking water. But you developed new technology, which is not written in that law. So to implement that technology, law should be revised. So to revise that law, we have to conduct the various examination to check whether it is safe to use. And then we have to change the law to permit use of the technology. And for example, if you develop a good ship, which have desalination facility on the ship, and then the ship can move to another island to supply drinking water. But in Korea, it is not easy to use that kind of ship because based on law, drinking water source should be taken a certain area, not the whole sea. So the ship should be go to the certain area to take the water and move to another area to provide the product water. So there are various limitations using the new technology. So we have to think about law before we use the technology. So law can be driver and can be hurdle for using the new technology. Okay, now let's move to the innovative water technology. I will show you some of the example of the innovation in first wastewater treatment, and then I will talk about membrane technology, and then finally I will talk about less water news. This slide shows the evolution of the wastewater treatment technology. At the first generation, people didn't consider about removal of pollutant. They just wanted to exclude rainwater or pollutant from their living area. They just wanted to exclude to another area at the first generation. And then people started to concern about the protection of the environment. So the new process to remove the organic matter has been invented. And then after removing the organic matter, people also interested in removal of nutrient, because one of the example of nutrient is nitrogen and phosphorus. If there is a high concentration of nitrogen, phosphorus, and you emit that kind of the wastewater to river, then there will be the algal bloom. So to protect, to reduce the algal bloom, we have to remove the nutrient from the wastewater. So now the technology status is around the third generation, and the fourth generation, we are started to think about extracting some valuable material or nutrient from the extracting that kind of the valuable element from the wastewater. We started to research it, and then for the fifth generation, we have to think about the hazardous material. There will be the potentially hazardous material in the wastewater. So we have to think about how to remove those chemicals from the wastewater. So let me talk about wastewater treatment a little bit more. Before that, I'd like to talk about the definition of the environmental engineering technology. Environmental engineering technology means the technology that can separate unwanted pollutant from humans. That is environmental technology. And also we want concentrated. We want to reduce the volume of the pollutant. And also we want to decompose the chemicals, maybe toxic chemical to the less toxic chemical. So, and also if it is difficult to decompose the pollutant, we want isolated from the humans. And this is the traditional definition of environmental engineering technology. And recently, the definition is extended. Environmental engineering technology also includes recovering valuable materials or generating energy from the waste. It is also belongs to the environmental technology. So there are many mechanisms relating to the water treatment technology. The first one is the separation, and second one is concentration, and third one is decomposition, as I mentioned just the previous slide. Relating to the separation, we can use the density property of the pollutant or size of the pollutant to remove this the pollutant from the water. Maybe sometimes we can use the electric charge of the pollutant to separate from the water. And adsorption and solubility can be used to separate, and also to concentrate the pollutant. Similar mechanism can be used. Density, size difference, and charge difference can be used. And for the decomposition, normally we use the bio oxidation or reduction process, or sometimes we use the chemical oxidation process. The example of the density difference can be the settling tank. Using the gravity, we can separate the particle from the water, and the size can be relating to the membrane process. And for the electric charge, there is electrodialysis or electrodeionization process. And for the solubility, coagulation and fluctuation process is belong to the using the different solubility. And for the biological oxidation process, that is kind of the how can I say that there is activated sludge process, which is widely used in the wastewater treatment process, as explained in the YouTube before this lecture. And for the chemical oxidation, we can apply the chlorine or the UV or ozonation to oxidize some pollutant in the water. This is the representative process of the wastewater treatment process. Here, we use the gravity to separate some big size particles. And then, as explained in the YouTube, we use the aeration tank. In the aeration tank, it is important because we grow the bacteria, and bacteria eat dissolved organic matter. Dissolved organic matter is difficult to remove by settling because it cannot settle down using the gravity. Then we have to convert the pollutant to something that can be settled down. So in aeration tank, dissolved organic matter is converted to the microorganism. Microorganism, the density is higher than the water. So after this process, we can separate the microorganisms from the water. So we can, that means we remove the pollutant, which is converted from dissolved form to the particle form. So that is one of the important principles of this process. So this is the mechanism what happening in the aeration tank. So we have the dissolved organic matter. Bacteria eat the organic matter because the bacteria want to grow or to increase the number of the bacteria. So it needs some carbon source to generate the cell, and also to generate the cell, it needs some energy. So by oxidizing the organic matter, it can generate energy, and also using the carbon in the organic matter, it can generate, synthesize a new cell. So the final product will be new cell, new bacteria, and also degraded byproduct, that is carbon dioxide and water. For this process, we have to put the oxygen. So this is called the aerobic condition. So using the aerobic bacteria, we can remove the dissolved organic carbon from the water. So from the after aeration tank, we can separate the pollutant as activated sludge, which is a bacteria. Then we have to also treat these bacteria. To remove some, this is also kind of the form of the organic matter. So getting this sludge, we do the anaerobic digestion. This anaerobic digestion process, the bacteria can be decomposed more, and we can remove the more remaining the organic matter, and also we can generate some useful gas, that is methane. This shows the mechanism of anaerobic condition process. And by using the methanogenic bacteria, we can generate methane. So in this process, bacteria also use the organic matter as a carbon source for the synthesis of cell, and also it oxidize for the energy production, and then finally it can generate methane. So in wastewater treatment plant, it can remove the pollutant and also it can generate some source of the energy as a form of the methane. Now I'd like to talk about more the biofuel. As I told you at the first part of this lecture, the European country can reduce the emission of the CO2 because they applied a lot of renewable energy. So this left graph shows the annual growth rate of the global renewable energy source, and the first length solar PV, solar power energy, and the second one is the wind, and the third one is on biomass. So biomass can be a kind of the renewable energy to reduce the emission of the CO2. But although the growth rate is the third among these renewable energies, if you look at the total world's major energy supply source, biofuel accounts for a large portion. For example, from the total energy source, coal and oil accounts for many part, and also there is a renewable energy, and among the renewable energy, biofuel accounts for around the 10%. So it accounts for more than solar power and wind power. So I think it is good to think about using biofuel. And one of the reasons why we have to care about the biofuel is this kind of the problem by the solar PV. Solar power plant may be not a environmentally friendly system. For example, in Korea, we have the mountain area region is around the 30%, and also when we build the new solar power plant, we build it on the mountain. That means we have to cut the trees from the mountain, then we install the solar power PV on the mountain. And when we have the heavy rain, like last week, we had a lot of the landslides problem, and also people claim that this kind of solar power plant caused severe landslides. So after this kind of the accident, we have to remove the solar panel, and also it is difficult to remove the waste of the solar panel. So maybe solar power plant may be not an environmental friendly system. So that's why we have to think about biofuel. I'd like to talk about the microalgae. Microalgae have high content of lipid. So because using the high content of lipid, we can generate biofuel. And microalgae is similar to plant. Using the sunlight, we can generate this the energy, and also it capture the CO2. Using the CO2, it can generate this, the cell. So the carbon source for the cell synthesis is CO2. So we can remove the CO2 using microalgae. Also, using certain temperature and retention time, we can easily grow the microalgae, and also it can be used to removal of the nutrient like nitrogen. And this character microalgae, we can microalgae for the removal, and this is the schematic of microalgae process removal of from the wastewater, and it can capture CO2 from the power plant. So that means that we can reduce the emission of the CO2. And various countries started the pilot scale test to grow microalgae in the US and UK and Japan. And one of the obstacles for the mass production of microalgae is harvesting process. To use the microalgae for the biofuel material, we have to at the first time cultivate microalgae, and then we have to harvest and then remove the water, and then we can use it. But for the harvesting process, the stage accounts for 20 to 30% of the total microalgae production cost. So to implement this technology, we have to develop economical process for the harvesting of the microalgae. And I think membrane process can be used for the microalgae harvesting process. I will talk about it later. Until now, I talked about the removal of the pollutant from the wastewater, and also I talked about the energy production from the wastewater. So we can generate renewable energy from the wastewater, and we can reduce the emission of carbon dioxide. And now I'd like to talk about a little bit more about the sensors. Previously, I talked about the Korean Ministry of Environment revised the law to change the index for the organic matter concentration from the COD to TOC. So there is a new need to measure the TOC measurement. And one of the problems is that the existing equipment for the TOC measurement is expensive, and also it takes around 30 minutes. 30 minutes may be short, but it's not a real time. So there is a need in low cost and real-time measurement of TOC in Korea. To measure some pollutant in real time, it is better to use the optical property of the pollutant. For example, there are various organic matter in the wastewater, and the organic matter has a unique optical property. So if we give the light at certain wavelengths, the organic matter absorbed the light, then emit at different wavelengths. We call it fluorescence property. Using this fluorescence intensity, we can estimate the concentration of organic matter. In addition, using the machine learning technology, we can improve the prediction of the concentration from the fluorescence intensity. This slide shows some example result obtained by my PhD student from Indonesia, UPI. Left graph shows the relationship between the measured TOC and the predicted TOC using the multilinear correlation of the fluorescence intensity, and right graph shows the result by machine learning. And the low value of the RMSE means the more higher accuracy. So as you can see this graph, we can tell that the machine learning technique can be helpful to obtain better accurate result. So I talked about the removal of organic matter and the recovery of the resources, some valuable elements from the wastewater. And finally, I'd like to talk about the removal of the hazardous material. Actually, there are many material in recently, like EDC and disinfection byproduct can be harmful to human beings, and also there can be some pharmaceuticals in the wastewater, untreated wastewater. So we have to think about these chemicals, and also there is some new material like nanoparticles, and also there are also the nano size plastic is a kind of the issue in environment. So these chemicals, some of them already known as very harmful, but some of these, the risk or toxicity is unknown. But to before we find the toxicity of these unknown chemicals, so we have to find, it's better to find the removal efficiency by wastewater process. Because we live in the city and use a lot of various chemicals, and then we emit the chemicals to the wastewater treatment plant. And if this wastewater treatment plant cannot remove completely these chemicals, then it will go to the river and it can cause some problem in ecosystem. And also maybe we can take the water to make the drinking water, and the drinking water should remove completely these chemicals. But some of these chemicals will be difficult to remove, then we can drink these chemicals. So it is important to measure what kind of chemicals is included in the river and water treatment process. I tried to measure some of these pharmaceuticals. These are the examples. Some are the caffeine and carbamazepine, diclofenac, and atenolol. These are widely used the medicine by the humans. And when I measured the concentration of these chemicals in the Samjin River, which is one of the biggest river in Korea, I detected these various materials in the river. That means wastewater treatment plant cannot remove these chemicals completely. And I found that high concentration of the caffeine, ibuprofen, and TCP, although this range is nanogram per liter level, but we have to maybe think about this concentration, because when we add all these chemicals, it can be to the microgram per liter range. So how to remove these micropollutants from the wastewater? So existing facility using biological oxidation system cannot remove completely, maybe. So that's why we have to apply another types of the process. One can be the membrane, because membrane have a different size, and the chemicals that I showed you have the molecular weight around 200 to 600 gram per mole. So if we use the very small pore size membrane, then we can remove those kind of the chemicals. But one of the obstacle of the use of the membrane is energy. By the definition, membrane is a kind of the selective barrier. That means selective means it can pass maybe water, and it can remove the pollutant by the membrane. And barrier means we have to put some energy for the transport of the water. So the energy is a kind of the important issue to implement this system. Actually, reverse osmosis membrane can be used for the wastewater treatment, and also recently it is widely used in the desalination field. At the in early 1950s, the thermal desalination technology has been widely used, but after 2000, almost 100% of the new desalination plant uses reverse osmosis technology instead of thermal desalination process, because reverse osmosis uses lesser energy compared to the previous thermal desalination process. So energy consumption is key to select best water treatment process. Let's think about how much energy is used in seawater desalination plant. Total energy is required around 3.5 kilowatt to produce 1 ton of drinking water, and around 74% of the energy is consumed at RO system. Because high energy is required at RO system, compared to the other pre-treatment or intake or disinfection system. The reason is we have to use a lot of energy for the high pressure pump. So to reduce desalination energy, we have to develop new kind of the technology. First one would be the improvement of high performance pump efficiency. And also we can make a better RO membrane, which can use less energy for the water production. And also we can develop a better energy recovery device, and also we can optimize the whole process to reduce the specific energy in the RO process. Then by developing this kind of process, how much can we reduce the energy in desalination? So more dynamically, ideal energy is 1.6 kilowatt per ton. So this is the ideal value. So we cannot reduce lower than this value. Then now, what is the status? Now the current status is 1.88 kilowatt per ton. It is only for the reverse osmosis part, not including pre-treatment and disinfection part. And including the pre-treatment and other processing in SWR plant, the real process, the energy consumption has been decreased around 3.9 kilowatt per ton. The energy consumption is different depending on the site of the plant and capacity. As you can see here, we improved the technology and the energy consumption has been decreased in SWR plant, but the technology has been almost saturated around between 3 and 4. So I think it is very difficult to reduce the consumption of the energy less than 3. That means we have to find new technology to reduce more energy from the 3 or 3.5. In Korea, we have conducted many research, desalination research, and one of the biggest research relating to RO was started from the 2006, and then we wanted to reduce more energy, so we approached or investigated a new process instead of RO. One of the examples is FO for the osmosis, and the another one is pressure retarded osmosis membrane technology. After investigating various technology using FO and PRO, we found that it is a little bit difficult to implement this technology, and then we moved again to the RO process again to reduce the optimize the process. And then after this period, recently we are investigating more new process to reduce the energy for the desalination. That is solar energy or desalination battery. That is a new kind of the research topic. From now, I'd like to talk about what is RO and FO and PRO. I think I used around more than 1 hour. Is it okay to continue? Okay. Let me think about explain about what is RO and FO and PRO. If two solutions are divided by membrane, one is a high concentration solution, maybe seawater, and the other one is fresh water, then we can have the transport of water by the osmosis phenomena. So low concentration of water will move to the high concentration of water. It is called osmosis, and also it can be called as a forward osmosis. And in case of reverse osmosis, we pressurize high concentration water part, and then we can have the fresh water. So this means we have to give the pressure higher than the osmotic pressure difference of these two solutions. And if we apply some pressure which is less than osmotic pressure of these two concentration, two solutions, then there will be still some flow of the water, but the flow will be less than FO, but will have the same direction. If we use the force and connect to the turbine, then we can generate electricity. That is the principle of the pressure retarded osmosis. So before I talk about the FO and RO and PRO, I'd like to talk about the osmotic pressure, how to calculate it. We can calculate the osmotic pressure using Bantoff equation, and to calculate the osmotic pressure, we use the mole concentration of ion in the water, and if we multiply the gas constant and the temperature, then we can calculate the osmotic pressure difference. I have a short quiz here. Can you calculate the osmotic pressure of the seawater if we assume that seawater has only NaCl 35 gram per liter and the temperature to 20 Celsius, and the osmotic coefficient is 0.95. Yes, okay. I will give you 20 seconds to think about this, how to solve this, and to get the to calculate the osmotic pressure. To calculate the osmotic pressure, we have to convert the concentration to the mole concentration using molecular weight of NaCl, and we have to think about the total ion in the water. NaCl will dissociate in the water 2 ions, so we have to multiply 2 from the mole concentration of the NaCl. So then we can calculate the osmotic pressure of the seawater. Then if you calculate it, you can calculate the osmotic pressure. The answer is 27.7 bar. So osmotic pressure of the seawater is around the 30 bar. Okay, 30 bar. To get some water using reverse osmosis process, so we have to apply more than 30 bar, right? So we can get water from the membrane depending on different pressure. If we apply a lot of pressure, then we can have more water, right? So as you can see in this graph, if we apply more water, then we can generate more water, right? The slope is the property of the membrane. But in case of the seawater, this is in case of the freshwater filtration. But now we have to filter seawater for the desalination process. That means we have to change the equation a little bit, because we sacrifice osmotic pressure for the production of the seawater. So if we apply around the 30 bar, which is the osmotic pressure of seawater, then we cannot generate the water. So we have to apply more pressure to generate or to have the product water. Then if we apply 10 bar or 5 bar, then the direction of the water will be opposite, right? And also if we don't apply any hydraulic pressure, that is called osmosis pressure on process, then we can have the negative water flux. So between this point and this point is a PRO process range. Okay, so this slide shows the schematic diagram of PRO. So if we put into the membrane the wastewater effluent here, and then if we put the seawater here, there will be osmotic pressure difference. Then the water will transport from the wastewater effluent to the seawater side. And also we apply certain pressure, then because of the water movement, we can turn the turbine and then we can generate electricity. So from the concentration gradient, we can generate energy using the PRO process. And I'd like to talk about FO process, which doesn't, when we doesn't apply any hydroelectric pressure, then that is FO. So this slide shows the advantage of FO. If we apply hydraulic pressure a lot, then the pollutant can be compact, then it will be difficult to remove. But in case of FO, there is low hydraulic pressure, so we can have the loose pollutant layer, so it is very easy to remove. So FO has a low energy demand, and also it is easy to remove the fouling layer. And in case of if we want to apply this FO system to the seawater desalination process, then we can use the highly concentrated water, which is called the draw solution. If this draw solution have a osmotic pressure more than 30 bar, then the water will move from seawater to this draw solution part. And then if we can recover fresh water using low energy, then this process can be efficient for the desalination process. This is another application of FO. We use high concentrated fertilizer, and here seawater is coming in, and because the fertilizer has a higher concentration, then the water will move to the fertilizer part, then diluted fertilizer can be used for the fertigation. And this is another example. And similarly, we can apply the fertilizer, and in this case, we can use the microalgae. As I told you, we need to consume a lot of energy for the harvesting microalgae, but if we can apply FO system for the harvesting of the microalgae, we can reduce the energy for the microalgae harvesting. And this is another application. And effluent is going in, and seawater is going in. Because of osmotic pressure difference, the water will go to the seawater part, and we can have the diluted seawater. And energy consumption is proportional to the concentration of the salt. So in the next step, we can apply the less energy for the desalination. That is also another application of FO. There could be various application. And finally, I'd like to talk about last section of my presentation. This slide shows the how much we use water in different countries. The average household water consumption is around 200 liter per person per day. So every day we use around the 200 liter in average. And in Korea, we use 180 liter per person for every day. So Korean consumes around the 90% of the world average. And we analyzed where we used this tap water. So interestingly, around 25% is used in the toilet, and it accounts for around 50 liter per person per day. So every day we use around 50 liter in the toilet. So if we can reduce amount of water in the toilet, then we can generate less amount of pollutant. Then we can reduce the energy for the treatment of wastewater. So that is a concept of the waterless toilet, and it is an important part of the Science Warden project conducted in Korea. Science Warden project was conducted by the university in Korea, UNIST, and the research period was 7 years, and the research funding was 7.6 million USD, and around 50 researchers participated in this project from the different university, research institute, industry, and private organization. You can know about more information from this website about the Science Warden project. The name Walden came from these two books. David Thoreau wrote this book. This book is the story of a man who lived alone on the shore of the Walden Lake and on self-sufficient base for the 2 years. And this book became the beginning of the environmentalism. And around 60 years later, famous psychologist Skinner published this book, Walden 2. In this book is about the community. First book was a man who lived alone, but second book was relating to community, where everyone can live happy. So it's relating to utopia. And from these books, the UNIST made the name Science Walden. This is relating to the community solving social problems using science. And this is a convergence of science and technology and humanity and arts. So the main goal, the final goal of the research was changing waste to the energy and by the natural life. So one of the important part of this project was developing new type of waterless toilet, and the name was BB. And as you can see, this the picture, the feces and urine was separated because generally feces can be degraded using the anaerobic process to generate methane. But if urine and feces are mixed together, then the methane production efficiency can be decreased. So that's why they separated the urine and feces. And from the feces, they can generate the biogas, methane, and then using the methane, they can use the fuel for the general life. And also, they extended this concept to the economic life. For example, if someone use this BB, the toilet in that community, then they can get some money. The unit of the money is wool, so they can get the 10 wool by using the toilet. Then using the 10 wool, you can buy some coffee. So it extended the fuel generation, biofuel generation, and also it extended to the normal economic life. So this is a kind of example of the wool exchange platform. So using feces standard money, they can exchange some product like coffee, and also they can buy some hat from this platform. So these are all I explained, prepared today. And also in the last part, I prepared some topics to share. That is, how can we be creative? Because I already used a lot of time, so I will quickly introduce this kind of the topic. The creativity, everybody I think want to be creative person, and also I want to be a creative person. And based on the book, it says that everyone has a potential to be creative. So creativity is a kind of form of problem solving ability. And also there is certain way you can increase the creativity. I prepared some ways how to increase the creativity. Some people think that I have kids, and the Korean people are interested in the growing the kids and very interested in the education. And we want many people want to teach various things to improve the creativity, like the arts and the physical education, like the music and other things. But I think to improve the creativity in engineering, this kind of the art, music, is not useful. I think to increase the engineering creativity, you have to increase the basic knowledge, like the chemistry, biology, and mathematics and physics. We have to have the various knowledge in various from the various curriculum in your university. That is one of the best way. And also maybe it is difficult to remember all the things, but try to remember the principle. And later, maybe 2 or 3 years or 10 years later, maybe you have to find some method to solve some problem, and maybe you can need some knowledge from the other classes. At that time, you can open the book and then easily follow the knowledge if you understand the principle now. And also we have to invest something to improve our to be an expert, that is time. So we have to invest a lot of time to be an expert in a field, and also concentrating in a certain topic is important to have the creativity. And there is certain ways to improve the immersion or to get into the flow state. So you always think about the goal or what you have to solve, and remove some distraction. That is the basic, and also do the exercise. I think it is an important thing. After exercising, you can reduce your stress, and then you can be more healthy. Then you can think about, you can have a more creative idea. And final one, yes?
You have 4 minutes left.
Yeah, I will use 1 more minute. Thanks.
Thank you.
And the final one is trees. Trees is a kind of the theory for the innovative problem solving. There are 40 principles to think about to solve some problems. Or I think you can have some problems, or sometimes you have to make some patent. At that time, you may have some difficulty to make new idea, but I think this can give you a certain hint. So please look at visit this website, what the trees are. And using the certain principle of this component, simple and composite membrane can be invented. Okay, these are a summary for my presentation. Global exchange, climate change is one of the most important issue in R&D of the water treatment technology for the sustainable environment. And also the water treatment technology have to meet the needs from the technical, economical, environmental, social, and legal needs. And the convergence of the technology between the environmental technology and IT can contribute the development of the innovative water treatment and for the better life. That's all for my lecture, and thank you very much for the attention. Thank you.
All right. Wow. Many thanks for Professor Lee for such a fantastic talk about Wastewater, and yeah, there's 100 slides and it cover issue about environmental issue all around the world, what drive innovative technology, as well as innovative technology development such as reverse osmosis and science Walden. Okay, Professor Lee, before we move to Q&A session, we will give you maybe a little rest to have some drink. So while we are waiting, the committee will show us a short video.
Wastewater. It's what gets flushed down the toilet, rinsed down the drain, and produced by places such as factories, workplaces, and homes. According to the U.S. Environmental Protection Agency, the average person uses about 100 gallons of water a day, with almost all of it turning into some form of wastewater.
Of the 100 gallons per capita per day of wastewater we produce, we are drinking about a gallon per day per capita. Essentially, wastewater is the 1 gallon of human waste produced per capita per day, diluted with 99 gallons of non-human waste.
Because wastewater contains pollutants and contaminants, most communities send it to treatment plants to be processed. The water has to meet federal, state, and local standards before it's released back into the environment. Wastewater is one part of the human water cycle, which describes the many ways people interact with nature's hydrologic cycle to meet our needs for water, food, and energy. While water treatment plants are a necessary part of public health and environmental safety, they require a great deal of energy and resources to operate.
In the US, we devote about 3 to 6% of the national energy budget on the enterprise of clean water.
Kartik Chandran is a professor of Earth and Environmental Engineering at Columbia University and funded by the National Science Foundation. He investigates new, more efficient ways to treat wastewater. Wastewater contains elements such as phosphorus, organic carbon, and nitrogen, which are often removed during the treatment process.
Well, let's start with nitrogen. When we want to remove nitrogen from wastewater, in addition to the organic carbon, the energy footprint of a wastewater treatment plant essentially doubles.
At the Newtown Wastewater Treatment Plant in Brooklyn, New York, treating wastewater involves several steps. First, the system separates floating matter and solids from the water. The remaining sewage then enters an aeration tank.
What we do in aeration tanks is we supply oxygen to microorganisms that actually treat the sewage.
Typical microbes for wastewater treatment require oxygen to degrade organic pollutants. The resulting sludge is further treated, and in its final step, the water is disinfected. Chandran is trying to reduce the amount of energy used in this process. He uses a type of bacteria found in wastewater called Anammox. These organisms don't need oxygen to remove nitrogen from wastewater. While traditional wastewater treatment often releases greenhouse gases such as nitrous oxide and carbon dioxide, using Anammox bacteria does not.
They allow us to remove nitrogen at far lower energy costs and chemical costs. Not just that, we would save 60% in terms of aeration costs if we use the Anammox model for removing nitrogen.
Chandran and his team are also looking at ways to use wastewater's abundant chemicals, nutrients, and other resources to produce energy.
We have been able to convert the organic carbon in food waste, in sewage sludge, in fecal sludge to biodiesel, which is a liquid fuel easily transported and easily used, directly used in diesel engines.
The Newtown Wastewater Treatment Plant is already producing energy from organic material.
The digesters are heated to about 95 degrees Fahrenheit. They act like the human body. They break down the organic material, and we provide no oxygen. And one of the byproducts of the digestion process is methane gas.
Methane gas is a common energy source.
We are right now using a good portion of our digester gas to heat our boilers at our locations. The boilers are used for heating the environmental areas, the personnel areas, and they're also used to reheat the digesters.
Chandran and his team are also finding additional uses for wastewater products. For example, they are using the organic carbon found in wastewater to develop bioplastics and taking the phosphorus and nitrogen from wastewater for use in irrigation.
By linking the water sector, the energy sector, the food sector, and a few other sectors, we are now able to address these challenges not just one by one, individually in isolation, but together in conjunction. That's a very sustainable model.
As Chandran and his team help make the human water cycle more energy efficient, they are also helping us see wastewater not as waste, but as a precious resource.
All right. Thank you for the committee for the break video. And right now we are in the Q&A session, but please wait because I think Properly has been there. Okay. we are in the Q&A session, all the participants. So for all participants who want to ask Professor Lee, you can write your question in the AHA slide. The link has been given to you in the chat box. Otherwise, you could directly ask the speaker. Please let me know by raising your hand, emoticon, and I will let you ask. But before, while we're waiting for the direct answer, so we move to a slide first. So Professor Lee, here we are. We have several questions from participant in a slide. The first question is biocomposite material is good absorption method to treat wastewater, or do we just focus on traditional method to treat wastewater? Please, Professor Lee.
Okay, thank you for the good question. And there is a kind of the material called the biochar. It can be natural or bio absorbent. Normally, it can be generated from the some fruit peel, or for example, banana peel can be turned into the biochar, and the biochar can absorb some the pollutants such as the iron and other the metal element that can be removed by biochar. So there is some research relating to the bio composite material, and also the coffee powder is also can be used to make the biochar. That is an interesting research topic. And one of the problem is we have to put some energy to turn the that kind of material to the biochar. That is the essential part. So if we can reduce the energy to production of biochar, then it would be the best way for the removal of the pollutant. Thank you.
All right, so basically biocomposite can be used as absorption material, right? Okay, we move to the next question from Satria. He is our student in UPI. Maybe Satria, could you ask Professor Lee directly?
Okay. Dear Professor Lee, what is your suggestion to us as students to make innovative technology, especially for wastewater treatment, so we can make environment better? 감사합니다.
Okay, thank you for the question, and also your pronunciation is very good. Well, very tough question. A student can try many different new topics. Maybe it is difficult to for the implementation with the low cost. But anyway, for the lab scale tester, I think you can try various things. For example, I suggested some pressure retarded osmosis membrane processor, and also I suggested some kind of the forward osmosis processor membrane. You can make that kind of the process in the lab, and also you can think about new types of the process. For example, one of the innovative way to invent new technology is a combination or hybrid of the process. So I think you can change some flow pattern, then you can generate a new concept for the treatment. That would be a good way. And also, similarly, and I think you can study many various the water treatment technology and think about how to combine two different treatment technology to get the merit of each process. That would be the good way. And also, I'm thinking about these topics always because it's kind of the how can I say, it's popular in Korea to make a new technology from their own background curriculum knowledge. So I also think about that, and it is actually not easy making this kind of thing. Thank you.
Okay, you're welcome. Thank you very much.
Okay, thank you, Satria, and also for Professor Lee for the answer. We move to the next question. Dear Prof. Lee, based on your experience, what is the hardest to remove material or contamination in wastewater? Yeah, material that are hard to remove based on your experience. Sorry, Prof. We can't hear your voice.
I'm sorry. Okay. Good question. Actually, the problem is not the removal efficiency. The problem is how efficiently or effectively remove the pollutant. For example, I think carbon activated carbon is a kind of the old process, but I think it is the best process which can remove the organic matter from the water. And also, if you use the membrane process, you can remove the almost all the chemicals from the wastewater, and also you can apply the oxidation. So you can combine the AOP, advanced oxidation process, and also membrane. Then you can remove all the pollutant from the membrane and from the water. But the problem is the cost. If you add more and more processes, then the price will increase, and that is that can be the answer. And also, the pollutant which can be difficult to buy the membrane. There is a certain that kind of the element which doesn't have any charge, and the length, it has a long length, not the spherical shape. If it has a long length, then sometimes it is difficult to remove by the reverse osmosis membrane. One of the example is a TCP, which is a fire retardant. I showed that in my slide, and that is one of the difficult chemical which can be difficult to remove by the reverse osmosis membrane.
Okay, thank you, Prof. Lee. Basically, all the contaminant, or yeah, we can remove all the contaminant, but the matter is the cost, right?
Right.
Yes. Okay, the next question. Dear Prof, I'm curious about the simple technology of water treatment that can be used to fulfill the water needs in the disaster area, since sometimes water scarcity occur after disaster. Please, Prof. Lee.
It is also a very good question. I think to use some water treatment process in disaster area, that means it should be operated without electricity, right? So actually, I have one experience. I made the membrane treatment process which can be operated without electricity. Instead, we combined some handle like using hand, the pump can be operated, or I connected with the bicycle shape. So if we turn the pedal, then we can the pump can be operated, and then using the pump operated by human power, the membrane could be operated. So I made that kind of the equipment, and I sent to the various countries like the Africa. But I didn't get the feedback from them, because sometimes that kind of membrane facility needs some kind of maintenance. So I'm curious about how long they used it. Sometimes I sent to the Vietnam and Mongol and Africa and many countries, but I'm curious about the feedback from them. Anyway, this is a very good question. Actually, I also curious about some kind of the good and simple water treatment technology which can be used in the disaster area. And also another example for the the treatment technology is using FO is there is a hydrolation bag. In the FO, that bag has some high concentration of the glucose. So because of the glucose, it has a high osmotic pressure. So you can put into the dirty or just the normal river water, then using the osmosis, the pack can be increased. So final product is diluted glucose. If you drink it, you can drink water, and also you can drink nutrient from the hydrolysis pack. It can be it already made in US, and it is for the digester area purpose and also the war area. So military can use it drinking water from the certain water scarce area, clean water scarce area.
All right, thank you, Prof. Lee. But I'm curious about the water availability in Korea after the flood disaster. Is there any problem about the water availability in Korea after the flood disaster?
Not much actually. Flood disaster happened last week, and we don't have a certain problem relating to the drinking water source, but yeah, not yet. But drought is a kind of the maybe important problem, because in certain period, like in May, beginning of May, we don't have enough rainfall. So in the industry complex need some water for the factory. But in that case, they suffer from the water shortage. But without that, from the flood damage, we don't have a certain that kind of the problem yet.
Okay, thank you so much, Prof. Lee. We move to the next question. How significant is the potential of resources from the ocean today and in the future in overcoming environmental issues such as using algae, sponge, and other resources? Can you show us example?
Okay, it's a good question. Actually, the ocean and desalination is one of the best way to have the stable water supply, because many people in the world live near the coast area. So that's why desalination is important. Yeah. So in the future, if the people doesn't have enough don't have enough water, then desalination will be the only way to provide sufficient and stable water supply. And overcoming issues, algae, sponges, is that the issue in the ocean? I don't understand exactly.
I think related to the global warming, because there is no connection with water availability.
Yeah, I think then my answer was enough to talk.
Okay, thank you, Prof. Lee. Next question. What is your experience with operating wastewater? Okay, I'm sorry, Prof. Lee.
At the bottom one, maybe?
Yeah. What other method can we use for desalination other than microalgae? And is the practical application of desalination is high due to maintenance and operational cost of desalination?
Actually, microalgae is not the desalination process method. Yeah, I think maybe I explained it not well. Anyway, but I'm interested in using the microalgae for the desalination in the future. Maybe next research topic will be the use of microalgae for the desalination. Not many people tried microalgae desalination, but will be similar like the biofuel cell. Biofuel cell technology has been applied or investigated for the wastewater relating to wastewater treatment, and also they mentioned about that they can generate some energy from the using the microfuel cell, and also I think using the similar principle using the microalgae we can use it for the desalination. I think that would be my future research topic.
Perfect. Thank you, Prof. Lee, for the answer. We move to the next question. This simple question. What is the alternative to desalination, Professor Lee?
Okay. So yeah, to have the stable water supply, I think the alternative is the reuse of the wastewater from the wastewater treatment plant. So water is cycling as a one water. So we use and we emit to the wastewater, and then almost the same amount will be produced from the wastewater treatment plant. Then if we treat it completely, then we can drink it or use it again. So yeah, so for the alternative, the desalination, the water reuse will be the answer.
All right. Next, in 2023, which method of wastewater treatment is best, cost-effective, environmentally friendly, easy to adapt, less experiment analysis involved, and universally acceptable? Professor, is suggestion again.
Yeah, I think normally the general or traditional method, which is the activated sludge wastewater treatment method, is widely adapted in the world, and it is cost effective maybe, but we want to reduce more energy, and we also want to generate energy from the wastewater treatment plant. So that is a kind of our future way to go. But anyway, yeah, activated sludge method is widely used in the world, and it is a good way, and it shows a good removal efficiency too.
All right, thank you, Professor Lee. Next question. Is the RO technology practical for household use?
You may have the in the house. You may have the water treatment machine. Normally it
I don't think
use
so.
the RO technology, right?
Yeah.
So yeah, so it can be used, and also in my house I also use the RO treatment machine for drinking water. Actually, at the first time I didn't want to use the water purifier in my home because I believe the water quality from the drinking water facility in Korea. But sometimes some pollutant can be emitted from the pipe. So I worry about some particulate matter from the pipe. So that's why I installed the water purifier in my home. And also, in case of the SWRO, seawater desalination RO, we have to put a lot of money and energy for that. But for the general use, kind of the freshwater treatment, we don't need a lot of energy. So it is a critical technology to use in the household.
Okay, yeah. In Indonesian, it's the same because in several homes we have air purification for water. Yeah, for installment. Okay. Next question. Thank you for the presentation. If we apply the forward osmosis process, is there any kind of byproduct that we have to consider? Thank you. Whether it is harmful or something like that.
Yes, good question. Byproduct, actually the final byproduct will be diluted draw solution, diluted high concentrated solution. That is a byproduct. Sometimes we have to extract some draw solute from the diluted solution. And if draw solute is toxic, there can be a problem. But in case of if we use the fertilizer as a draw solution, then diluted solution can be directly used for the fertigation. So we don't have to worry about the recovery of the draw solute. Actually, one of the obstacle or important part is what is the draw solution for the FO system. Yeah, we have to select that best draw solution for the process that will determine the economical efficiency of the process.
Okay, thank you, Professor Lee. We move to the following question. What is your experience with operating wastewater treatment equipment? Please give us example of a time when you had a troubleshoot problem with a piece of equipment. Thank you.
Actually, I don't have experience treating the wastewater treatment equipment yet. But I had experience in the drinking water treatment equipment, and at that time I applied my reverse osmosis membrane to investigate the removal efficiency and performance of the membrane process. And when I visit the drinking water treatment plant, I think when we operate that kind of the big pilot system, many unexpected things happen. So in the lab, we can handle the problem easily, but in the pilot scale test, it is difficult to handle because the size is big. And also, if we want to change a simple parameter of the process, it may be difficult if the size of the pilot system is big. So yeah, I think that's my experience. So similar to that, because the size of the real facility is too big, so maybe it is difficult to change a little bit parameter of the process. That would be an important part.
Thank you, Professor Lee. Next question. Dear Professor Lee, how do we measure how good an RO membrane is? Is there some specific criteria that have to be fulfilled?
Yeah, RO manufacturer provide some kind of the data sheet to show the performance of the membrane. For example, they show the NaCl removal efficiency, and also they sometimes provide the boron removal efficiency to show the data for the RO. So I think normally the NaCl removal and boron removal is widely used, and also when applied certain pressure, they measure the flux from the membrane. So flux removal efficiency is important factor for the RO membrane.
Thank you, Professor Lee. Maybe we just have two last question, I think. Okay, the last two questions, Professor Lee. Thank you for the amazing lecture. Can we use wastewater from industrial waste? How different is the treatment for wastewater which sourced from the seawater and industrial?
Yeah, the source and the component are very different. Yeah. For example, some wastewater from the paper company, it has a lot of the sulfate in it. So we have to think about how to deal with the high concentrated sulfate. And also we have to think about how to deal with the final waste from the wastewater. So I think different factory will produce the different characteristics of the wastewater. And depending on the characteristic, we have to choose a proper way to remove or apply. And also seawater, it has a high concentration of the salt. So that is main difference between the wastewater and seawater.
All right, thank you for the answer, Professor Lee. We have a very active participants, so there are many questions. So, in your experience, which is the best between organic and inorganic adsorbent-based material used in filtration membrane? Which one better?
Which is the best? For the membrane processor, we need to put some pre-treatment. If there are a lot of the organic matter, sometimes we put the carbon dioxide. No. The activated carbon before the membrane process, because activated carbon efficiently remove the organic matter. So yeah, I think the activated carbon will be the best pre-treatment method. And I don't know about the inorganic absorbent material.
Yeah, the last question, Professor Lee. What types of microalgae species are usually used for wastewater treatment?
Actually, I don't remember the exact name of the species. I think Bulgaris, yeah, microalgae, yeah, was one of the example, and there was some species in the research paper, but I don't exactly remember the species. I'm sorry about that.
All right, it's okay, Professor Lee. Well, it's all. All right. Again, many thanks for Professor Lee for the fantastic talk and clear explanation for all questions that has been given. And we almost arrived at the end of the second session, and we really appreciate the knowledge given by Professor Lee from Kyungpook National University, South Korea. Furthermore, on behalf of Department of Chemistry Education, we would like to extend our gratitude to Professor Lee for your precious time and availability to give us many insight, especially in wastewater treatment. For that, please receive the certificate of appreciation for becoming a speaker at the virtual summer school 2022 Green Sustainable Chemistry for a Better Future. Once again, for all attendees, please give praise and appreciation for Professor Lee. Thank you so much, Professor Lee.
Thank you. Thank you very much.
Okay, before we move to the next session, maybe is there Bu Fitri as the chairman of Virtual Summer School? Yeah, Bu Fitri will give us some question, some word.
No question. It's already the question. All right. Thank you, Mora. Very good afternoon. Probably, yeah, it's really glad and nice for me to seeing you again after last year. Yeah, if I'm not mistaken, we invited you as the one of invited speaker in our world class professor program. So again, on behalf of chemistry department, I would like to extend our thankful for your great effort and contributions to our Virtual Summer School program. So today, finally, we can, how to say, provide a variety of knowledge for our participants according to the sustainable development goal issues, yeah, especially regarding to the clean water, yeah, and in this moment, you share with us many important and crucial informations, especially in water innovations technology that I do believe should be contribute a lot, yeah, in how to say, in providing clean water for our society. And this is one of the target of this program. Probably we would like to enhance, to encourage our participants to be more aware, more concerned with the surrounding, yeah, especially about water. That this issue also, I guess one of the biggest issue, not only in Indonesia but also over the world. Yeah, everyone really curious how to provide the clean water for our daily life. So hopefully, by the knowledge and also information that just been shared by properly, participants can earn more knowledge about that and also improve their awareness how to use water, how to say, more wisely. Yeah, since yeah, it takes a lot of cost if we have to do treatment later. So again, Profi, thank you so much for your very comprehensive and excellent lecture today. I really do enjoy your. Thank you.
Okay, thank you, Bu Fitri, for the remarks. And maybe before we stop this session, from Professor Lee, do you have any closing remarks for the participant?
Thank you very much for the active participant. I didn't expect that in this kind of various questions, but yeah, it's very impressive to have many questions, and hopefully my presentation or lecture is helpful for you. Yeah, thank you very much.
Once again, thank you so much, Professor Lee. And before you left us, we will have a photo session. So for all participants, it's time for the photo session. Please, if you don't have any connection issues, please let your camera be open, and my fellow committee will help us to take the photo. Okay. Okay, Bu Trianisa will help us to take the photo with Professor Lee.
So, okay, everyone, if you don't have any trouble with your camera, please turn on your camera so we can get your picture. Okay, I will count in 1, 2, 3 for slide 1. Okay, for the next slide is 1, 2, 3. And for the final slide, 1, 2, 3. Okay, thank you very much. Give it back to moderator.
Yeah, thank you, Putri. Once again, thank you so much for Professor Lee for the nice talk, nice explanation, and hopefully we can meet again in the next occasion. So thank you so much.
Thank you, Prof. Lee.
Thank you.
Wish you a good day. Yeah.
You can leave us if you have any other business, Professor. Thank you.
Okay, thank you very much.
All right, for all the participants, as usual, we still have a quiz session, yeah, that will assess you about the material that has been given by Professor Lee about wastewater innovation in wastewater. So the link for the quiz still ongoing, be published by committee, but we still use AHA slide, yeah, like usual. Okay, the link has been put in the chat room, slide.com/IEACT. So please, ladies and gentlemen, please enter the slide. All right, 8 players have been join us. How many soal kan? All right, 32 players have joined. I wonder where is Priya? She don't attend this day. Okay, I think the number is remain constant. So shall we start the quiz right now? Okay, the quiz will be start in 3, 2, 1. What does TOC stand for? This is an easy question. But don't confuse with C. What is C? Alright. Total organic carbon. The second question. Okay, congrats for Anissa. Yeah, the contender winner, I think, Anissa. Number 2, economics needs of wastewater treatment innovation in operating aspect is move to the third question. And Anissa still lead the line. The highest country water stress is Qatar, Israel, India, Uni Emirat Arab. Okay, of course, Qatar, yeah, the host of World Cup 2022. Okay, Siti Rodia now is the leader. Number 4, here is the third generation wastewater treatment, except to the fifth question. And now we have the new leader is Dihara. Here is the social needs for innovative environmental technology except energy efficiency, environmental friendly, aesthetic needs, or automation. Answer is energy efficiency. We have two last question, and right now Zahrina is the leader, followed by Rosita and Shafira. Okay, what factor should be a concern in the separation and concentration in the main mechanism for water treatment? Density, size, electric charge, density, size, solubility, density, size, absorption, or density, size, chemical oxidation. Alright, the answer is density, size, and electric charge. Shavira now is the leader. Okay, one last question. Yeah. What is the material from microalgae material that can be used as a biofuel? Who's the winner in this session quiz? Okay, the answer is Philippines. And who's the winner for us in this afternoon? Here is Zahrina. Congratulations, you win. Oh, the last. Okay, okay. What is the main problem in the mass production of microalgae? This is the very last question. All right, harvesting. See, Zarina win the quiz. Congratulations, Zarina, the new contender. Okay. Well, now we are at the end of day 4. All participants, time flies so fast, isn't it? So everybody, don't forget to attend tomorrow's session because we still have many prestigious speaker, and don't forget that tomorrow we will start in at 8 AM, yeah, with 4 speaker, yeah, and yeah. And also, I would like to announce you about the single figure presentation submission that the deadline is this night. Yeah, so please don't forget to submit your SVP to Padlet. The committee will resend again the link to you. Yeah, so don't forget that the deadline is this night Indonesian time. Again, here we are, the information from Ibu Mita. Yeah, as you know that Bu Mita is the student of Professor Lee, as well as the lecturer staff in UPI. If you want to see more information about Professor Lee Research Group, you can visit the laboratory website. Yeah, you can just click and you can have the contact of Professor Lee. Yeah. All right, for the committee, is there any other announcement? Enough? So it's time for attendance link. Yeah. Okay. For all participants, please don't forget to fill the attendance link so we know that you are attend the activity. Yeah, because it's connected to the credit. Okay, here we are. We have sent you the attendance link in bit.ly/FSS 202227. Yeah. All right. Thank you so much for all participants for your actively participation here in the fourth day. There will be one last day tomorrow, so please don't miss it. Yeah. Okay. Enough ya. Enough ya. So thank you so much for today, all participants, and see you tomorrow at 8 p.m. Bye-bye.
Thank you, everyone.
Located in the green premise of Universitas Pendidikan Indonesia, Bandung, West Java, the Department of Chemistry Education is one of the oldest departments in UPI, initially established as the Natural Science Program in 1954. The Department of Chemistry Education occupies Japanese architecture Jika building, whose name depicts strong collaboration with the Japan International Cooperation Agency. We provide an academic atmosphere for a satisfying experience in studying chemistry and chemistry education. Aligned with the UPI's vision, the Department of Chemistry Education under the Faculty of Mathematics and Natural Sciences Education is determined to be a leading and outstanding department in chemistry and chemistry education. The Department of Chemistry Education strives to prepare professional chemistry teachers and chemists who can compete globally, also establish and strengthen national, regional, and international networks. These missions are supported by 7 professors and 40 academics, consisting of 24 lecturers holding a doctoral degree, and 16 lecturers are prospective doctoral candidates. The Department of Chemistry Education offers 4 study programs, including Bachelor of Chemistry, Bachelor of Chemistry Education, Master of Chemistry, and Master of Chemistry Education programs. 3 study programs have been accredited A by the National Accreditation Council for Higher Education. This rank indicates high qualification and recognition of study programs in fulfilling the national education standards. For Master of Chemistry program as a new study program has met the minimum requirements for accreditation. The 4 study programs are now undergoing the accreditation process by the International Accreditation Board. With the existence of educational and non-educational programs, the Department of Chemistry Education implements the cross-fertilization principle to build synergy between the 2 programs and strengthen the 3 pillars of higher education that consist of education, research, and community service. The curriculum structure in the Department of Chemistry Education is designed to prepare students either for professional work or for graduate studies. At the bachelor level, students will be studying fundamentals of chemistry, analytical chemistry, physical chemistry, inorganic chemistry, organic chemistry, biochemistry, applied chemistry, lab experiments, and additional course of chemistry education for students of the educational program. While at the master level, students will be exposed to a more comprehensive knowledge and research of chemistry, chemistry education, and their applications. The Department of Chemistry Education is supported by 4 expertise or research groups in the chemistry program and 7 in the chemistry education program to excel at the 3 pillars of higher education. The chemistry program specializes in material chemistry, environmental chemistry, biological chemistry, and food chemistry. Material chemistry group focuses on the transformation of biomaterials or waste materials into nanomaterials, porous materials, and hybrid materials that can be used as catalysts, corrosion inhibitors, adsorbents, and membranes. Environmental Chemistry Group conducts research on bionutrients, biohydrogels, membranes, thin films, bionanomaterials, nanocomposites, and modeling studies.